Okay, good
Buenas tardes a
afternoon,
todos.
everybody. It's one
Es
o'clock,
la
so
una,
I'm going to try and
así que
keep
voy a
us
intentar
on
que
time.
respetemos
My name's Graham
el
Lipkin. I'm a
horario. Yo
kidney
soy
doctor looking
nefrólogo
after an adult
y me
cystinosis
ocupo de una
clinic
clínica de
in
cistinosis
Birmingham,
en
in the UK,
Birmingham, en el Reino Unido. Me llamo
and I'm
Graham
going to
Lipkin
be the
y
chair
presidir
for
esta
this
sesión.
session. This
Es una
session is
sesión
one of
que
the
constituye
highlight
uno de los
sessions
puntos
of the meeting.
más importantes de esta
I
reunión.
mentioned
Esta
this
mañana ya he
morning that
mencionado que
we have
tenemos
had a fabulous
Hemos celebrado una
scientific
sesión
session
científica fabulosa. Fue
yesterday
ayer
of clinicians
con
and of
clínicos,
scientists, and
científicos,
so
etcétera,
we're going to
y
give you the
vamos a
highlights of
presentar
that
las
meeting
conclusiones más importantes
today,
de esta
things that have a
reunión y
particular
cuestiones que
immediate
tienen
impact
repercusiones
on
inmediatas en la
patient
atención a
care.
los pacientes.
So the
La
organization of
organización de
the
esta
session
sesión
is we'll have
consiste
two
en lo siguiente.
talks. The
Escucharemos dos
first two
charlas.
are
La primera
focused
se
on
centra
both
en
clinical and
aspectos
experimental
clínicos y
aspects
experimentales
of the
de
prodrug
el
CF10,
fármaco
after which we'll have a short
CF10. Después habrá
question and
una sesión de
answer
preguntas y
session,
respuestas y
and
luego
then we'll move
pasaríamos
on
a
to 4 more
4 charlas
talks
más y
after that, and
después
then have another
habrá
question
otra
and
sesión
answer.
de
So
preguntas y
after
respuestas.
the
Después de
first
las
two,
primeras
there are roving mics.
2,
Please feel free to ask
pasaremos
questions.
unos
You're very
micrófonos
welcome.
inalámbricos para que
We'll
puedan
try
hacer
and
ustedes sus
answer them
preguntas
for
e
you.
intentaremos
So
responderlas.
it's a real
Es
great
un placer,
pleasure
pues,
to introduce
presentar
our
al primer
first speaker,
orador,
Professor
el
Herbie Newell. Many of
profesor
you will know Herbie.
Newell. Muchos lo
Herbie is the emeritus
conocen, ya
professor
es
of cancer studies at the University of
profesor
Newcastle and the
catedrático
professor of
en la
drug
Universidad de Newcastle y
discovery
catedrático de
at
descubrimiento de
University of
fármacos en la Universidad de
Sunderland.
Sunderland.
And Herbie
Además,
has very effectively and dynamically taken on the mantle for the development of
recogido el testigo del
CF10
desarrollo del
after
CF10 con
the
mucha
very sad
eficacia
loss of
tras la
Rosalind
triste
Anderson.
pérdida de
So,
Rosalind
Herbie,
Anderson.
thank you
Así
very
que
much.
muchísimas gracias. Herbie, tienes la palabra.
Thank you very much indeed, Graham, and thank you all for the invitation to come and speak to you today. Earlier on, we were asked about one word that you would use to describe this meeting and this community, and the word that came straight to my mind was inspirational. You really are cystinosis patients and ill families and carers a real inspiration to all of us involved in biomedical research. And for me personally, it's been a phenomenal honour to be able to inherit, as Graham said, under tragic circumstances, the CF10 project from Ros Anderson, and to realise this project not only as a much better therapy for cystinosis patients, but also a lasting memorial to a truly great lady. So I'm going to give you an update on where we are with the development of the cysteamine prodrug CF10. And Eleanor very kindly earlier on gave you a brief insight into this, but I'll give you a bit more detail. So you will be aware, painfully aware, of the drug cysteamine. It's a very useful drug. It's actually, of course, the only proven effective treatment for cystinosis at the moment. It works by removing cystine. It's a very simple and relatively cheap oral medication, and it has actually a very elegant mechanism of action, as we heard earlier on this morning. But, and of course there is a massive but, it's an absolutely terrible drug to have to take in terms of the side effects, which are listed here, and I won't reiterate them because you live with those side effects day in, day out. Probably the most important concept in pharmacology, and definitely in drug development, is the concept of therapeutic index. It's the ratio between an active dose and a toxic dose. And for a good drug, that ratio should be big. So the dose you need to take to treat whatever problem you have is much, much lower than the dose which would cause a side effect. And so if you've got a headache, and I was a very good boy last night and the generosity of
para no
Cystinosis
sentir los efectos
Foundation
secundarios.
UK
Yo ayer
at the dinner
anoche fui
provided us with
me
excellent
porté
food
muy
and
bien.
drink, and I
Ayer
moderated
me
on
moderé
drink,
mucho
so I didn't need to take any
con la
paracetamol this
bebida,
morning.
a
But
pesar
if
de
I
la
had
generosidad
taken a
de
couple of
los
paracetamol,
organizadores
it would have
de la
worked,
cena,
and I would know
que
that
fueron
for
muy
that
generosos.
paracetamol
Si
dose to
hubiera
have
tenido dolor de
been
cabeza, me hubiera
dangerous,
tomado un par de
I would have had to
paracetamoles
have
y
taken an
si
awful lot more than the 2 tablets that would control
el
the
fármaco
headache. And
hubiera
so
sido
it's got a
peligroso,
very
pues hubiera
big
sido
therapeutic
distinto.
index.
No hubiera podido tomarme
I know
dos,
through
sino mucho menos.
talking
Sé,
to
por hablar
patients,
con
carers,
pacientes,
and
padres
parents here that
y
essentially
cuidadores,
no
ningún
patient
paciente
taking
que
cysteamine
tome
has no
cisteamina
toxicity. There are always
carece de
side
toxicidad.
effects.
No
And
todo el
so
mundo tiene
the
efectos
best thing we
tóxicos
can
y
do
efectos
with
secundarios. Así que lo que
cysteamine
podemos
is
hacer con la
consign it to
cisteamina
history,
es
and that's what we're trying to
dejarla
do.
para la historia y es lo que estamos
And the late
intentando
Ross
hacer.
Anderson
Y
came
Ross
up with a
Anderson
really elegant
buscó
way
una
of
manera, encontró
approaching
una
this
manera muy
problem,
elegante de
and that's to design what we call a
enfocar este
prodrug. And a
problema, que es
prodrug
un
is a chemical which
profármaco.
is
Un profármaco
inactive
es
in
un
its own
producto
right,
químico que es
but
inactivo,
after
pero
you've
después de
taken
tomarlo
it and it's
y
gone
después de
into
entrar
the
en el
body,
cuerpo, se
it's
convierte
converted to
en
the
la
active
especie
species.
activa, en el
And
producto
this
activo.
is
Este
the
es el
prodrug
profármaco
that Ross
que
developed.
desarrolló
So it
Ross.
has
Y en
in
rojo
red
contiene
the
el
active
fármaco
drug,
activo, que es
cysteamine.
la
It's
cisteamina, y está
attached to
unido
a
a un grupo
glutamate group
glutamato
which
que
targets that cysteamine to
lleva
the
la
tissues,
cisteamina a los
and that's
tejidos,
shown
y eso es
in
lo que
blue.
vemos en azul.
And then
Y
2
luego
other
hay dos modificaciones
modifications are
adicionales que
needed.
también se necesitan.
First
La
of all,
primera
there
es
is
el
an
grupo
ester group
éster,
shown
que
in
está en
light
azul
blue,
clarito,
and
que
that's to
sirve
cover up
para
the SH
recubrir
sulfhydryl group in
el grupo
cysteamine,
SH
which
sulfamina
because
de
it
la
is exposed in
cisteamina,
cysteamine,
que si está
gives the
expuesto en la
problem of
cisteamina provoca
irritation,
irritación
the problem of
y
ulceration,
úlceras
which
que
indeed
en
can
algunos
sometimes
casos pueden
progress
acabar progresando
through to
hasta una
perforation,
perforación,
actually
porque
holes in the
acaba
gastrointestinal
provocando
tract. But by covering up that
perforaciones
with an acetal
en
group, you prevent that
el
problem.
sistema
And
digestivo,
lastly,
pero con
it
el
has
acetol
an ether
se
group.
puede
Sorry, that was
eso
the
era,
green
perdón, eso era
thioester
el
group.
tío
Lastly, it has an ester
el
group,
verde,
and that is to
y
make
luego
sure
también
it's
tiene un tío
absorbed
éster
really
que
effectively from the
se asegura
gut.
de que se pueda,
The
perdón,
key
el tío
thing with
éster
a
era el que hace
prodrug
de
is it
cubrimiento y el
handled
éster nos
in the
asegura la
body the way
absorción
it
en
was
el
designed to?
intestino.
And so the
La
first
primera serie de
series of
experimentos que
experiments we
nosotros
did
hicimos
are what we call metabolism and
fueron para analizar el
pharmacokinetics.
metabolismo
This
y la
is looking
farmacocinética, es decir,
inside the body after you've given a
ver qué hace el
drug
cuerpo
to
después de
see
haber
what the parent drug,
dado un
CF10,
fármaco.
has
¿En
been
qué se ha
converted
convertido
to.
este
And in our
profármaco que es
case,
el
critical
CF10? En nuestros
conversions
casos, la
are,
conversión
first
más
of all,
crítica es
to
primero
remove the thioester
que
and the
se
ester groups,
desaparezcan el
and
grupo
then at
tioéster
the
y éster,
tissues
y
to
después que en los
remove
tejidos
the
se
glutamate group.
elimine el
And the
glutamato.
first series of experiments
El
which
primer
Roz
tipo de
did before she
experimentos que
died
hizo
was to
Esther
look at
antes
this
de
metabolic
morir,
process
Ross antes de morir
in
fue mirar el
vitro.
proceso
So that's
in vitro,
in a
es
test
decir, en
tube in the
un
lab. This is
tubo de
before you've got
ensayo,
as far as doing
antes
any
de los
animal
estudios
studies.
animales, en el laboratorio.
And so what Roz
Lo que
showed was that there are
Ross
2
mostró
enzymes we
es que hay
call
dos
esterases
enzimas, las
that very
esterasas,
effectively
que
clipped off the ethyl
quitan
group and clipped off the
el tío y
acetal group.
los grupos
And then
éster y
there's
tioéster,
another
y hay otra
enzyme
enzima,
called
la
gamma
gamma
GT
GT, que
that
está
is present
presente
throughout
en el
our
cuerpo,
bodies
en
on
todas
every
las
cell
células que
that clipped off the
también
glutamate group. And
desencadenan
that
el
enzyme is on
glutamato
the
y se encuentra en la
surface of the
superficie de las
cell.
células. Por
So
tanto,
the
la cisteamina
cysteamine would be carried in the
se
blood and
transforma
extracellular
por la
fluid
sangre
to
y los
cells
líquidos
throughout the body,
extracelulares hasta las
and
células del cuerpo, y
then
entonces
it
allí
would be
se
cleaved off by loss of the glutamate group, and you would deliver
desintegra, se divide,
that to the tissues
porque
where the
se
cysteamine is
corta
needed.
el grupo tiol,
So
y
that was
entonces
the
allí
theory,
se entra
but
en
is there
la
evidence
célula. Pero esa era la
preclinically that
teoría.
that's
Luego, ¿qué
the
pasa
case?
clínicamente?
And this
En esta
first
primera
graph
gráfica
here
nos
shows
muestra
the
los
levels
niveles de
of
cisteamina
cysteamine and this
y
critical
su
intermediate gamma
intermediario tan
glutamyl
importante que
cysteamine
es la
in the blood of rats given
gamma
CF10 orally.
glutamilcisteamina en
And
las
the line at the
ratas a las
top
que se les ha
is the
administrado, ratas con
intermediate gamma
cistinosis.
glutamyl
Y arriba
cysteamine at
vemos los
very
niveles
high
muy altos
levels,
de la
and down the
gamma
bottom are the
glutamilcisteamina, y
levels of
abajo
free
vemos la cisteamina
cysteamine
libre
in the
en
blood.
sangre.
And what we
Nosotros lo que
wanted were
queríamos
high
era
levels of the intermediate and low levels of free
niveles bajos
cysteamine.
de
And that's
cisteamina
because by
libre.
having low levels of
Si hay
free
poco
cysteamine,
cisteamina libre,
you
se
prevent the
impide que la
cysteamine having all the
cisteamina
nasty
tenga todos
side
los efectos
effects that
secundarios
you
tan
get when you
feos que
take
tenemos cuando
cysteamine
se toma
directly.
directamente. Y
And
eso
that's, of course,
es lo que
here, as you
vemos
can
aquí.
see,
Esto es exactamente lo
exactly
que se
what
está
we saw.
consiguiendo. Poca cisteamina
So this is
sangre.
great.
Y eso
It was
está
behaving
fantástico porque
metabolically as designed,
metabólicamente se
but
comportaba tal y como
critically,
se había diseñado.
does it
Pero luego,
actually
¿qué?
deliver
Luego era capaz
cysteamine to
de llevar la
tissues? And so
cisteamina
we use the
a
kidney,
los tejidos.
obviously a very
usamos el
important
riñón, que es un órgano
organ
muy
in
importante en la
cystinosis
cistenosis,
to
para
answer that
estudiarlo
question.
y responder la
And
pregunta.
this
Y
shows
aquí
in
vemos
the
arriba
top line is
la
the concentration
concentración
of
de
cysteamine
cisteamina
in
en
the kidney,
el
and the
riñón, y
bottom
abajo
line
vemos la
is the
concentración de
concentration
la
of
cisteamina
cysteamine in
en
the blood.
sangre.
And
Y
then
luego aquí otra
here
vez
again you can see is a
vemos una
very big
diferencia muy
difference.
grande.
I
Hubiera
should
tenido
have
que
pointed it out in the
decirlo ya
previous
antes en la
slide
diapositiva
that the
anterior.
y-axis
El
going
X de la,
upwards is
el
a
X de
log
la
scale.
Y
So there's a
es una escala
massive difference
logarítmica,
between
es
the
decir, que hay
cysteamine
mucha
in the
diferencia
tissue
entre la
where you
cisteamina
need
y el
it
tejido
and
donde
the
se necesita, y
cysteamine in the
la
blood
cisteamina en
where
el
you
tejido
don't want
y la
it.
cisteamina
And
en
that
sangre,
again,
que
as
es
I
donde
say,
no
is
queremos que esté, y
exactly
eso es lo
as
que
Ros
había
designed this
diseñado Ross
molecule to behave.
cuando diseñó esta molécula.
So that's
Hasta aquí,
great. It's
fabuloso.
handled
Se gestiona
metabolically, okay,
metabólicamente
exactly
tal
as
y como estaba
designed,
diseñado.
but
Muy bien,
critically,
pero
does it
¿sigue
still
funcionando
work?
o no?
And
Antes de
before she
morir,
died, through many
con muchas
collaborations with
colaboraciones
this
con esta
community,
comunidad,
Ros
Ross
generated
generó
some cell line
algunas
data and very limited
datos
data in a mouse
de
model and
líneas
some
celulares y
zebrafish
algunos
data.
poquitos
But
sobre modelos con
recently,
ratón, pero
as you've
como
heard
han
already,
oído
some
ya,
excellent
ahora
new
hay
models
modelos
have
muy
become
nuevos
available.
que
And we've
están a
had a
nuestra disposición, y
fantastic
tenemos una
collaboration
colaboración
with Jen
fabulosa con
Hollywood,
Jen
originally
Hollywood,
while she
cuando
was in
antes estaba
Auckland
en
and now
Auckland y
that
ahora
she's
que
back
vuelve
in
a estar en
Ireland in
Irlanda, en
Cork,
Cork,
looking
y
at
ha estudiado
CF10 in her
el
rat model.
CF10 en su modelo de
And this
rata.
is how
Este
that
modelo
model
se genera
is
de la
generated.
siguiente
It's
manera.
so-called
Se
gene
llama
editing,
edición
and it's produced
genética
a
o
model
génica. Edición
of the
génica.
disease which mirrors
Crea una
extremely
especie de
closely
espejo
the clinical
de la
progression
enfermedad,
of
modelo de la enfermedad
cysteamine and the
que
clinical
refleja mucho,
cystinosis and
muy
the clinical
bien
features
las
of
características
cystinosis.
clínicas de la cistinosis y su
And so,
conducta. Utilizando este
using that
modelo,
model, just in
resumiendo,
summary, looking
y
at
viendo
all
todas
of
las
the characteristics
de la
of cysteamine, which we
cisteamina,
know
que
well,
también conocemos todos,
we've
nosotros nos
asked,
preguntamos
is
si el
CF10
CF10
as good
funcionaba también
as
como la
cysteamine?
cisteamina, y la
And the
respuesta
answer
es que
is yes
sí,
in every
en todos
case.
los
So in
casos, en
terms of
cuanto a la
ability to
capacidad
deplete
de
cystine,
eliminar cistina,
in terms of
en
ability
cuanto a la
to
capacidad de
prevent
prevenir un
excessive
exceso
water
de consumo
consumption,
de agua
prevent
y un
excessive
exceso
urine
de producción de
production,
orina,
the incidence of
los,
Fanconi
de la
syndrome,
incidencia
the
del síndrome de Fanconi
histopathological and
y
pathological
los
effects on
efectos
the kidney,
patológicos e
and then the
histopatológicos
overall effect on
en
weight
el
gain in
riñón
these
y el
animals.
efecto en la ganancia de
The
peso en
two
los
drugs,
fármacos.
when
Cuando
they're
se
given
dan
at the
los
same
dos fármacos a dosis
equivalent dose,
equivalentes,
which
lo
we
que
call the
llamamos
molar
el
equivalent dose,
dosis
are
equivalente molar,
equally active.
tienen la misma
And
actividad.
as indicated
Y
in
como se
the
dice aquí
bottom text
abajo,
there, because CF10, as I'll mention in a moment, is so much
ser
less
tan menos
toxic than
tóxico el
cysteamine,
CF10 que la
we're optimistic that by increasing the
cisteamina,
dose, we may be able to get
esperamos
even more
conseguir más
activity.
actividad
And that's something that
pudiendo
we're looking at
aumentar
at the
la
moment.
dosis. Es algo que estamos estudiando
So
ahora mismo.
this is,
Y
apart
aparte
from
de
these most recent in
de los
vivo
datos in
data,
vivo
where Ros got the
más,
project to at the time,
recientes,
she was unfortunately diagnosed with a very aggressive
por
uterine
desgracia le
sarcoma.
diagnosticaron a Ross un sarcoma
So
uterino
as
muy
I took the
agresivo,
project over, the
pero ella
first
estaba
thing that
por
we
aquí,
did
y cuando yo
was
asumí
to
el proyecto
apply
tras la triste
to
muerte
the
de
Medical
Ross,
Research Council
fue
for
lo primero que hice
funding
fue pedir
to undertake
financiación
the
al Medical
regulatory
Research
preclinical safety
Council para hacer
studies
los
that you
estudios
have to do
preclínicos de seguridad
before
que hay
you
que
can
hacer
take
antes
a
de poder
compound
llevar
into
un
clinical trials
compuesto a
nowadays.
los ensayos
And
clínicos.
Ros
Y
was
Ross
able to
consiguió
apply
pedir
for
ese
that money in
dinero
her
a nombre
name
propio
before
antes
she
de morir,
died.
y
And
la
the
semana
week
antes
before
de
she
morir
died, she
recibió una
received a letter
carta
from the Medical
del
Research
Consejo de
Council.
Investigación.
And I'll choke here because it always
Estos que
upsets me
siempre
so
me
much.
molesta,
That
que se
her
decía que
project
su
had been
proyecto
funded in full to the tune of
se
1.6
financiaría con
million pounds, and it
1,6
was the
millones
highest
de
ranking project
libras,
they
el
had seen in
proyecto
the
de
last
más
5
fondos
years.
de los últimos
And
5
so
años. Así
Ros,
que
bless
Ross
her, left
se
us
fue
knowing that the project was going to go
sabiendo que el
forward.
proyecto saldría
And
adelante.
with that
Gracias a esta
funding, we were
financiación
able to
conseguimos
undertake a very
realizar una serie
comprehensive
muy
series
completa
of
de
studies.
estudios.
It's a legal
son una
requirement in most
exigencia
jurisdictions
legal,
that you have safety data
porque
in
tienes
a
que tener primero
rodent
datos de
and a
seguridad de una
non-rodent
especie de
species. And
roedores
industry
y
standard
otra especie
is
de
the dog
que no sean roedores.
and the
En
rat.
general,
And so
en
we
este
followed
sector
the
son
required
ratas y
procedures.
perros. Hemos
Had we
seguido los
not done
procedimientos y
this,
si no,
we
no
would
hubiéramos
not have
podido
been able to subsequently make CF10
poner
available
a
in all
disposición CF10
jurisdictions
en
around
todos
the
los
world.
países del mundo.
I
Así que
appreciate
entiendo
people
que hay
have very
muchas
strong
personas que
views about the
tienen
use of
muchas
animals in
reservas
biomedical research,
respecto al uso de
but
animales
this was a go, no go,
en
non-negotiable
la investigación
point.
médica,
So
pero
these studies are
esto
all
era
undertaken under the strictest
excluyente.
animal
Todo se
welfare
hace con unas
conditions.
condiciones de
They're
bienestar animal
unfortunate necessary
muy
step in the drug
estrictas y
development
son, por
pathway.
desgracia, uno de los pasos
And the
necesarios para el
results
desarrollo
were nothing short
farmacológico.
of spectacular.
Y los resultados
So
eran
in the dog,
espectaculares.
where
En el
the
perro,
drug is
en
given
que
as a
se
capsule
da
formulation,
el fármaco
which will
como
be the
cápsula,
initial
esta será la
clinical way
primera
of giving
manera de dar
CF10,
CF10 en
we
formato
didn't
clínico.
see
Aquí no
anything
se
at
observó
all
ningún
at
efecto
doses up to and
en
including including
de hasta 200
200 milligrams per kilogram for 28 days. If we went above that, we did see some minor signs of gastrointestinal toxicity, nausea and vomiting. But these are truly industrial doses. Critically, and this is a really important point and a result I was so pleased to see, if you look at the half-life of this key intermediate, gamma-glutamyl cysteamine, in the blood that is carrying the cysteamine to the tissues all around the body, the half-life is 4 hours. That's really important because it means that you won't have to take the drug more often than twice a day. And when we get to humans, you might predict the half-life will be a little bit longer still. So there's a very good chance this will be a once-a-day medicine. We did studies in the rat as well. The no adverse effect level was 333 milligrams per kilogram. This is at least 4 times higher than the dose that was as active as cysteamine in the rat model that I showed you the data for at the moment, a few moments ago. So we have a drug here that is as active as cysteamine, but has, we would predict, absolutely no toxicity. And no adverse effect means no adverse effect. So specifically, there will not be any hematological toxicity, no effects on your blood counts. There'll be no effects on clinical biochemistry, so no effect on liver function, for example. And we could detect nothing pathologically when tissues were removed and looked at under a microscope. This will, of course, be a lifelong therapy, and so we need to prove that there is no risk of genetic damage because that could lead to cancer. Now, there's no reason to believe this would be the case because cysteamine does not produce a significant increase in tumour rates, we know, and the other components all of us in this room take all the time. There's glutamate, well, we all take monosodium glutamate every time we have some soya sauce on our Chinese meals. There's an acetyl group, well, that's vinegar. If you've ever had fish and chips, you'll have had some of that. And then there's an ethyl group, which is coming, of course, from ethanol. And last night, I freely confessed already, I did consume some ethanol. So there's no reason to believe that there's going to be any toxicology in that respect. And then critically, you also need to look at the heart and look at respiration. Again, completely clear. So we were really, really reassured that there would not be a problem with the compound. Now, we're very aware, of course, that a major issue for cystinosis patients taking cysteamine is the problem of the unpleasant body odour and halitosis, which brilliant studies by colleagues in Leuven demonstrated are due to 2 metabolites of cysteamine. And so particularly methaneethiol and dimethyl sulfide are produced in the body from cysteamine after you've taken cysteamine. And so we wanted to know whether or not CF10 could produce these metabolites, because we would hope that by keeping the blood levels of cysteamine very low, we wouldn't see the production of these metabolites at significant levels. The data on the next slide are a bit complicated, but that just reflects the challenges of measuring the levels of these volatile gases in the breath of dogs. So in our dog toxicology study, we looked at a group of dogs who were receiving the no adverse effect level, so a dose that was completely non-toxic, 200 milligrams per kilogram per day for 28 days, and compared that to a control group who were not receiving CF10. And we looked at the animals on minus day 7, so that was before they'd had any cysteamine in this group here. On day 1, before they had any CF10, not cysteamine. And then after 28 days dosing, again, this is the control group. And this is the treated group. And these are 6 animals. And what we're looking at is the range of the levels that we detect. And you will see in brackets, if we look at the controls, let's look at these controls on day 1. These animals have had no CF10 and won't ever get any. Most of the animals actually had no detectable
no
levels
tenían
of
niveles
this
detectables de
very
estos
smelly
metabolitos
metabolite. But
tan
one animal had a
olorosos
very
como
high
el
level.
dimetilsulfuro,
Where did that
pero un
come
animal sí
from?
que tenía
Well,
unos niveles
any of us who have ever
muy
kissed a
altos.
dog
Si
will know
han
that
besado
kissing
ustedes
dogs
a un
can be
perro,
a
ya
little
saben que a veces es
unpleasant. Dogs
desagradable.
have
A
bad
veces
breath
tienen mal
occasionally.
aliento los
And
perros.
so
Y
that's
es
what this
esto,
is. It's
precisamente.
just the normal gut
Son las
bacteria of
bacterias
the dog
del intestino del
that
perro
by
normales y
digestion
corrientes
or
que por la
metabolism of the
digestión,
food
los
is
metabolitos
generating
de los
some
alimentos
noxious
generan estos
compounds.
compuestos nocivos.
However,
Pero
critically,
si
if we
vemos
look
este
at this
grupo de
group
aquí,
here, there is
aquí
absolutely no
observamos que no hay
difference
la
in
menor
the
diferencia en
exhalation
la
of
exhalación
dimethyl
de
sulfide after
dimetilsulfuro
28 days
después
dosing at a sizable
de
dose
administrar
in
durante 28
comparison to
días
the untreated
una dosis
animals or the
importante
same group before treatment. So the same
en
data
relación
are
con el
true
grupo
for the other
control.
metabolites. So
Por
we're
tanto,
pretty
tenemos plena
confident
confianza de
that
que
there is
no
not going to
habrá
be a body
olor
odor and
corporal ni
halitosis problem
halitosis
with our
con
drug.
este fármaco.
So you've
Eso ya
seen
lo han
this
visto
already.
antes.
This is where
Aquí es
we
donde
are.
estamos.
Rosie's, I was really
Me ha
pleased
encantado
to hear
poder
from
escuchar
Professor
al
Emma
profesor
yesterday
Emma
that at this
ayer
meeting
que
in
esta reunión de
2006, Ros
2006
presented
en
the
esa reunión
design
del
of
2006 Ros
CF10.
presentó el
And I know
diseño
it's 18
del
years later, but it's
CF10
great that it's
y
here. We're
aunque
talking
sea
about
10
it
años más tarde, es
transitioning
fantástico
into the
ver que
clinic.
estamos llegando
And
ya
so
al
Ros
momento de
described the
llevarlo a
design
la
and then
clínica.
the
Ros
discovery of
hizo el
CF10,
diseño y el
and we're now
descubrimiento del
right on the
CF10 y ahora
cusp of
estamos
going into a clinical
entrando
study.
en los
So
inicios de los
what we're
estudios
doing,
clínicos. ¿Qué
firstly, we're
hacemos?
doing
Primero,
some more
unos estudios
studies in
pequeños
Jen's
en
excellent
el
rat model
excelente
with higher
modelo de Jane
doses to see
para
what
ver
that
qué
shows.
significa todo esto y
And then
qué
we're going to
implica, y
undertake
después
a
haremos una
single
dosis
low
baja
dose
única de
CF10
un
pharmacokinetic
estudio
study
farmacocinético
in
de
cystinosis
CF10 con
patient
una única dosis
volunteers.
baja
And the
en
reason that we're
voluntarios
going to do that
pacientes
is
voluntarios de
exactly the
cistinosis.
same
¿Y por qué
reason
lo
as
vamos a hacer
I
así? Por la misma
gave
razón
when we did our first rat
ya di
study,
cuando
and that
hicimos el
is
primer
to
estudio con
prove
ratas para
that the
demostrar
drug
que
is handled by
este
your bodies
fármaco
in the
el
same way
cuerpo
as
lo
it's been
asimila
handled by the
y el
rats
cuerpo de los
and the
seres
dogs in
humanos
our
pues lo
preclinical
asimila igual
studies. Because
que
if
las
that is
ratas en
not
los
the
estudios
case,
anteriores, porque
then
si
we
no
need to go
tendremos
back
que
to the
volver
drawing board.
al
We have
inicio,
no
no
reason to
tenemos
believe
por
that it won't be
qué
the
pensar que
case,
no sea así,
but we
pero
do
hay que
need to
demostrarlo,
prove
claro.
that. And that
Y esto
has a number of significant
tiene una serie de
additional
razones
reasons. It's not
adicionales,
only
no
good
solamente es
science to do it
porque
that
la
way,
ciencia así lo
but
exige, sino
ethically
porque
it's
además
really
éticamente
important because
es
we'll mention in a
importantísimo
minute
porque
the initial
la
phase 1 and phase 2
los
subsequently
estudios
clinical trials
subclínicos fase
will
1 y 2
require a
exigirán un
major
compromiso
commitment
muy
from
grande
you as
de
cystinosis
los pacientes de
patients to take
cistinosis
part in
para
them.
participar
And
en
we
ellos y no
don't want to
queremos
ask you
pedirles a
to
ustedes
make
que se
that
comprometan a
commitment
participar
unless
a
we're
menos
really
que tengamos plena
confident
confianza de
that
que
the
este
drug
fármaco
is working in the
funciona
way it's
tal y como se
designed to
supone
work.
que tiene que funcionar.
There's
Por otro lado,
also an
también hay un
issue
tema de
of
logística.
logistics. A big phase
Programa
1
de fase
program
1
in
grande,
probably
seguramente
multi-centers as this
múltiples
will be,
centros
is going to take
sanitarios,
some serious
implicará una
organising.
organización
There's
bastante
also going to be a
estricta
financial
y también
driver for
habrá
doing this because the
una
phase 1
motivación
program
financiera.
is probably going to
El
cost
programa
best
de
part
fase 1
£5 million,
fase 1,
whereas
perdón,
this
seguramente nos
initial
costará como
study
5
is going to
millones.
cost about a
Este
quarter
primer
of a
estudio
million.
inicial, un cuarto
If
de
we
millón.
relieve
Así que
£5
si
million from the funding
conseguimos a
agencies,
las agencias de
it
financiación
means that that money is not available for
cuarto
other equally important areas of
de
biomedical
millón,
research.
pues no
So we want
podremos
to be
dedicar
really
ese dinero a
confident
otros
when we
ámbitos
ask for that money.
de
And not
inversión
coincidentally, but
en sanidad. Así
importantly,
que primero,
having
antes
these
de gastarnos
data
5
will
millones,
really
tenemos que
increase the
asegurarnos
likelihood that
de
we'll get
que
that
lo que
money.
hacemos esté bien. Y además,
So
si no
this
nos
study will,
aseguraremos,
as I say, be
si se
3
aprueba esto
cystinosis
y funciona
patient
bien, que
volunteers, a
tenemos más
single dose
probabilidad
of up to
de
3
que nos den la
100
otra
milligram
beca.
capsules,
Aquí vemos,
blood sampling over
pues
6
este
hours,
estudio se hará
analysis
con
of
3
the blood
pacientes
samples for
voluntarios de
these
cistinosis,
two
hombres de más de
critical
18 años, con una
components.
dosis
And
única
we
de
are
3
now at
cápsulas
the
de
end of the manufacturing of the
10
material, which has to be to a
se harán
special
análisis de
quality
sangre
standard
cada 6
called
horas,
Good
también habrá
Manufacturing
análisis de
Practice
muestras
or
de sangre
GMP.
buscando
We're
el
just about to
gamma-glutamina,
start making that
etcétera,
drug into
etcétera. Y la
capsules,
concentración
and
de
then
tiamina,
the
estamos ya a
patient
punto
treatment
de
and the sampling will be
empezar
possible.
con
We
el tratamiento con
hope that
pacientes y
we'll be
análisis
all ready
de
to go by
muestras.
the end of the
Esperamos que a
year
finales del año
and
ya
treat
estemos allí
the first
y que se pueden
patients at
empezar a
the
tratar los
beginning
pacientes a
of
principios
next
del año
year.
que viene. Estamos ya
There's a
fabricando,
lot of
etcétera, etcétera.
stages in making this happen.
Esto
The
implica
funding
muchas
has to
etapas,
come
la
from
financiación
somewhere,
tiene que venir
and
de
I'd
alguna
like
parte.
really to
Quisiera
acknowledge Sunderland
dar las
University
gracias a
here,
algunas de las
who have put their
universidades
hands in
que
their
se han rascado
pockets
el
and
bolsillo
provided
para
£250,000
proporcionar ese cuarto de
to
millón
make
de
this
libras
happen
para
quickly.
acelerar todo este
We
proceso.
could have gone off and different written grant applications, which would have gone to and throw lots of questions to answer, all of which would have taken time. But Sunderland, to my mind, very admirably took the decision. Let's cut to the quick. Here's £250,000. Go for it. The synthesis, as I say, is well underway, nearly finished. Manufacture's about to start. There is, as we heard in the earlier discussion, a massive amount of paperwork to be gone through to make all of this happen. Legal agreements, clinical protocols, lab manuals. We have to find someone to sponsor the study, take ultimate responsibility. And I'm really pleased to say it's ticked to all of those. And then we need ethics committee approval, and that's the last thing that we need to do. And then patient treatment and sample collection can start. So what does it actually look like when you make a drug for administration to patients for the first time? So on the slide there is a photo of colleagues at the High Force Research in Durham who are making it. In case you're wondering why they're all dressed up in what look like hazmat suits, that's not because the drug is dangerous and they're protecting themselves. It's because they are protecting the drug from themselves. We're all covered with bugs and other things that you probably don't want to look at under a microscope. They can fall into a drug while you're making it really quite easily if you don't cover yourself up properly. So that's what that's all about. And the formulation will be done at RVI Specials in Newcastle. The treatment of the 3 patient volunteers will be done by Graham at Birmingham, and then the analysis in a contract research lab. And so we are very, very nearly there to start that. In terms of a schematic, what that looks like is patients who volunteer to take part will be consented, will carry on taking their usual cysteamine dose, and then 2 days, 48 hours before the CF10, they'll stop taking the cysteamine to allow it to wash out of their system completely. Then we'll give the CF10, take the blood samples for 6 hours, and patients will go straight back onto their normal CF10 dose. And we are confident that that brief window without cysteamine, 48 hours, will have no significant impact on the management of one's condition. So please rest assured about that. The next steps after this will be a pilot run-in where we're going to do all of the laboratory investigations that will be part of the phase 1 trial of CF10, but in patients who are taking cysteamine. So this is just to sort out all of the logistics, because when we get to the phase 1 trial of CF10, we don't want to be developing and validating assays. We want to know that everything is going to work smoothly. So again, we will be asking for patient volunteers for this study, and we've had really great interaction with the patient community, the cystinosis community, in putting this protocol together, which again, the study will be done in Birmingham. And then once we know that everything logistically is going to work, we'll be ready to start the phase 1 trial, which we're anticipating will be, we hope, at the beginning of 2026. We've already sent an outline application to the Medical Research Council for funding. They loved it. £4.6 million. They're really keen to get in our back pockets. We now just have to put this final PK data from the pilot study in and submit the full application. And what this study will look like, there'll be 2 components. The first component will be 7 days of treatment. And again, we will consent patients with cystinosis to take part. It's really important to emphasize the reason this is being done in cystinosis patients and not healthy volunteers is measurement of cystine levels is absolutely critical to knowing whether or not we've got the right dose of our prodrug CF10. And you can't measure cystine levels in healthy volunteers because there aren't raised levels, of course, in healthy volunteers. So patients will stop taking their normal cysteamine. They'll then not be treated for 7 days to allow the cystine level to rise a little bit. So we'll then treat for 7 days with CF10, and we will gradually increase the dose of CF10 until we start to see the cystine levels come down. So we know we've got an effective dose. After 7 days, patients will return to their normal cysteamine dose. And then when we know we've got a dose that works, i.e. depletes cystine, we'll then swap to 28 days dosing, because once you have got 28-day exposure, you're then able to move on to continuous dosing, which is, of course, ultimately how we'll use CF10. And again, we may look at the dose level here. to make sure that we're giving a really effective, non-toxic, completely safe dose. So what's this going to mean for you as cystinosis patients and for cystinosis patient carers and parents? Well, we hope this will be a palatable and easy to take medicine, that there will be reduced side effects, specifically damage to the stomach and the gut, and the problems of bad breath, halitosis and body odor will be resolved. We should have improved tissue cysteamine delivery and hence a lower chance of kidney failure and extrarenal effects, with a reduced need for dialysis and transplant. Fewer doses should be required because of this increased half-life that we're seeing, and overall there should be an improved quality of life with knock-on effects of reduced psychosocial issues. And then lastly, overall, this will be a better treatment because there will be a reduced risk of failure because it will be taken more consistently. And all of this will be delivered due to the foresight of the wonderful Ros Anderson. So in conclusion, go back. Forward and forward again. In conclusion, just to note the current project team working on this, I'd like right up front to really thank Cystinosis Foundation UK, not just for their continued support, but for the fantastic foresight that they had in supporting Ros over many years to the tune of that £1 million to take CF10 from a concept to a clinical entity. And for a rare disease that the pharmaceutical industry would never invest that type of money in for drug discovery, it's, I think, an outstanding testament to the commitment of Cystinosis Foundation UK to generating a better medicine for this condition. So thank you, Cystinosis Foundation UK. We've then got the team at Sunderland University that I work with, the team that we are now working closely with at Birmingham under Graham to actually take this into patients for the first time. And then there's a huge number, an army of people out there working in contract chemistry companies, making CF10 in safety and toxicology studies, doing all this animal testing, the colleagues at RVI Specials at Newcastle, and then colleagues at Sundara Pharmaceuticals, who will ultimately, when this becomes a licensed drug, be responsible for marketing it. I estimate that at the moment, working on CF10 to get it to you, there are probably about 100 people in these different organisations. And so I'm told that we may have somewhere in the region of 220 cystinosis patients in England, and so probably half that number are beavering away in labs, bringing this new treatment to you. So I really hope that you feel that there is hope coming in the future. And then for people who outside the Sunderland current collaboration, people who worked with Ros previously, many of whom, of course, are in the room in various different areas. And again, I'd really like to highlight the work that we've done and are doing with Jen Hollywood on her brilliant rat model. And then last but not least, the funding bodies. So on taking this on, I was, of course, very pleased to help Ros at a terrible time in her life. And that, I will tell you, I'll be honest, has been a little bit of a responsibility on occasions. But I only need to come to a meeting like this for half an hour to realise it's worth it a million times. So thanks for listening to that. And we are nearly there. Next time, we hope we'll have clinical data to show you. Thanks very much.
Fabulous talk, Herbie. You are the man, and we wish you Godspeed in further development of this. Before we take questions, I'm going to invite the next speaker up because the two talks linked together. You've already heard a little bit about Dr. Jennifer Hollywood. Jen is got a PhD at the University College Cork, and then left us for a decade to go to Professor Ellen Davison's lab in Auckland. Thankfully, she's now returned to this side of the world, and she has been involved in developing animal models and using those animal models for future drug development. So it's a real pleasure to invite Jenny to come up and talk, and we'll then have a question and answer session with Herbie and Jenny. Thank you.
Thanks. Oh yeah, it's perfect. So yeah, thanks very much for the organisers for inviting me here today. It's really good to be here in person because the last couple of times I had to do online Zooms and things like that, so it's really nice to be here. So I'm going to talk today about some preclinical studies we're doing in the rat model that Herbie already mentioned. And as you know from Herbie's talk, cysteamine, and I'm sure you all are experts in this, cysteamine, although it does have advantages of depleting cystine, it does have many disadvantages. So one of the big problems is addressing these side effects. And thankfully, I think Herbie and the team have nailed that side of things. But another aspect of cystinosis that we need to address is the fact that cystine depletion is not the only thing we need to look at, because we know that taking cysteamine, even from birth, the patient will still progress to kidney complications and other complications. So there's something else going on that we need to look at and treat. So what we were doing in Auckland was modelling cystinosis using induced pluripotent stem cells. And these cells are like embryonic stem cells in terms of the fact that they have the ability to become any cell type of the body. However, they are derived from an adult person. So we take a skin cell, a biopsy, and then we reprogram these cells by culturing them in a dish and adding 4 genes, and then these cells reprogram back into a stem cell. And what happens then is you can coax these cells to become any cell type you want. So you can make heart tissue, lung tissue, but what we're interested in obviously is kidney. So we took these cells and we added a couple of factors, and they transitioned into what we call embryoid bodies, and then they further transitioned into these little kidney organoids. And these are mini kidneys in a dish, so they have all the cell types of the kidney that you need, such as the glomerulus, the nephrons, and the proximal tubules. And this is a really nice model then that we can study, and it's human tissue, so it's relevant. So what we did was we took these stem cells and we used a gene editing tool called CRISPR, and we knocked out the cystinosin gene to make cystinotic stem cell models. And then we coaxed these cells to become kidney organoids. So we had these kidney tissues. And by looking at these stem cells and kidney organoids, we found that as well as increasing cystine levels, having lots of cell death and large lysosomes, they also had this defect in a pathway that's very important for a cell. And Stephen spoke about this earlier, but I'll just go through it again. So in your cell, which you can say is your house, you generate rubbish, and you need to get rid of that rubbish. So you have these little garbage trucks, and this is what an autophagosome is. So it goes into the cell, or it's in the cell, and it sucks up all the proteins and the old parts of the cell that it doesn't want anymore. And then it transports them to the recycling center. And the recycling center is the lysosome. So the lysosome then will break down the components in the garbage truck, and then it will recycle what's useful to the cell back into the cytoplasm to be reused. And what we found was there was as well as having cystine accumulation inside the lysosome, they also had a defect in the autophagy pathway. So they were unable to fully recycle these components. And this block was causing the cells to become sick and other host problems. So we looked at being able to induce this pathway. And one way to do that is by inhibiting mTOR, which you heard about earlier as well. And to do that, we use this drug called everolimus. Now, everolimus is already a drug used in immune suppression and also in treating different types of cancers. So it's FDA-approved drug, and that was one of the reasons we chose it, so we could repurpose it. And when we combined this with cysteamine, we found that we could reduce or rescue all the cystinotic phenotypes or the symptoms in these cell models
métodos
and the
habituales
organoid
vimos
model.
que podíamos causar
So this was really good
cambio en
proof
los
of concept
mecanoides
showing
renales.
that in a human tissue
Esto
we're
lo
able to
vimos en
restore
tejidos humanos
the
y vimos
cell,
que podía restaurar la célula
the way of the cell back into a healthy
y se
cell
convertían en
type.
células sanas.
So we wanted then to test this
Por lo
treatment
cual
in a relevant
decidimos
animal model.
probarlo con animales.
So we produced our own rat model of
Producimos
cystinosis,
nuestras
again
ratas
using gene
de
editing techniques, which
cystinosis
Herbie talked
con
about.
edición
And
génica.
this rat model turned out to be really good
Y
in
la
that
verdad
it
que fue un modelo
modeled the
muy
human progression of disease very
satisfactorio.
well. So you can see
Vemos
here
aquí
in
en
this
este
graph,
gráfico
this is cystine
que tenemos
levels,
los niveles de
and this is
cistina,
the age of the rat. So at 3 months we started our
la edad, y vemos
measurements,
cuando empiezan en la edad
and
de
it
las
went from
ratas.
3 to 12 months.
Y
And you can see
vemos
in white, the white bars here are the cystotic
que las
rats,
ratas
and the black is the
sistemáticas
wild
son las
type.
blancas
And they had
y las
markedly
ratas
more cystine
naturales
accumulating in their
son las que
kidneys.
salen en negro.
And this is a picture of a
Y vemos
lysosome
aquí
under electron microscope, and this is the
los
crystal
cristales
when the cystine forms crystals. That's
a
what it
través
looks
del
like.
microscopio, las cristales de
And
cistina,
then we measured things for
con lo cual
Fanconi syndrome, such as
medimos
urine
todos
output. So
los
again, in gray here is the
outputs,
cystinosis
outputs de
animals,
orina, por
and in
ejemplo.
black is wild type.
Vemos que
And you can see that from 3 months
a
onwards,
partir de los
they were
3
progressively
meses,
urinating
la
more
progresión de la
compared to the wild type
urina
tissue
de
mates.
estas ratas
And we
cistinóticas era
measured
mucho
things
mayor
such as total
que las
protein,
ratas
glucose, and
normales.
calcium in these
También
rats. And
medimos
again,
la
we saw that these were
proteína,
all
la
increasing as
glucosa
time
y
went
el
by,
calcio. Vimos que
showing
en
that
las
they were
ratas
losing these nutrients in their urine.
cistinóticas, obviamente esto también
And then at
iba
dissection,
hacia
we saw
arriba.
that the kidney itself was markedly
También nos
different in
fijamos
the
que
cystonic
los
animals
riñones eran
compared to wild
diferentes. Si
type.
vemos aquí las fotos, comparan el de las ratas
So we did this
cistinóticas
preclinical
y las
study
ratas normales.
in Auckland for the
Así que en
past number of
Auckland llevamos a
years,
cabo
and I'll just talk you through the study design.
estudio.
So these are the
Este
rats
es el
here.
diseño del estudio. Estas
They're
son
Sprague-Dally
las
background rats, so they're
ratas.
white with red eyes.
Son de este tipo que tienen los
We gave them the
ojos
drugs in these little
rojos.
jelly pills because usually when you give a drug to an animal, you have to give it either
de
force feed
gelatina,
it or give it in drinking water, which isn't very accurate.
Con agua no
So
nos
we
parecía muy
devised this
adecuado.
jelly pill
Así que
system
utilizamos
so that
este
it was a lot
sistema
more
gelatinoso, este
palatable
formato
for the rat.
gelatinoso.
And because we were giving them
Y como
daily
eran dosis
doses, it's not nicer than
diarias,
giving them oral gavage.
es también mucho
So
más
cysteamine,
fácil
we
hacerlo
chose this
así.
dose.
La
It's
cisteamina,
a much lower dose than what
elegimos una dosis
patients would receive.
más baja
And
que
that
la
reason
de
for
las
that was because we
personas
wanted to
humanas,
see any additive effects
porque
of the second drug. We didn't want it to be like totally wiped out by
no
cysteamine.
queríamos
And we gave this twice daily. So every
que la
morning at
dosis
6
fuera
a.m.
mayor.
and 6
Y
p.m.,
dijimos
these
2 veces
rats
por día.
received a jelly pill of cysteamine.
Vemos aquí en la foto
And
cómo
then
toman
another
esta pastilla
cohort of
gelatinosa.
rats received everolimus.
Después
So they
teníamos el
received 10
everolimus.
milligrams per kg
La dosis
twice
era 10
weekly. So
miligramos por
every
kilo por dosis,
Monday and every Wednesday they
2
got an
veces a la
extra jelly
semana.
or a 1 jelly
Luego la
everolimus.
combinación también.
And then the next cohort
Que
of
el
rats
siguiente
received both treatments. So
brazo,
they
el
had
siguiente
cysteamine
cohorte,
morning and night, and then
una
every Wednesday
combinación.
or Monday and Wednesday they got an extra jelly
2
of
días a la
everolimus.
semana tenían
And
las
then we had
gelatinas
our
y
control
2 días
group,
tenían
and they
el
just got vehicle jelly.
everolimus.
So that was
Y
just
después
jelly with no drugs.
teníamos el
So these
brazo de
guys were getting
control que
this
solo
drug
tenía
for 6
gelatina
months.
sin nada
So we
dentro.
started at 6 weeks of age, and we went to 6 months.
Esto empezaba a las 2
And at monthly
semanas
time points, we collected
y
urine,
duraba 6
blood,
meses.
and we
Hacíamos,
took their
por
body weights
supuesto,
weekly.
controles de peso
And then at
a
the
las
end, we did these analysis to
semanales
see if there was any
y de
improvements.
orina y de niveles de cistina.
So first off, I'd like to show cystine
En
depletion.
cuanto a la
So
reducción,
in black here is
eliminación de
vehicle,
cistina,
cysteamine on its
vemos
own,
aquí en
everolimus
negro
on
es
its
el
own in blue,
vehículo,
and then the combination in
cisteamina en
purple.
rojo, en azul,
And just
everolimus
this is
y la
showing here the
combinación
amount
de
of cystine in the
tratamientos
kidney
en
tissue.
lila.
And you can see
Si lo
compared to the
comparamos
animals that received no
con los animales que no
drugs,
recibieron ningún
cysteamine reduced it by about
fármaco,
50%,
hubo una
but the
reducción del
combination of
60%,
everolimus and
pero
cysteamine
la
together had
combinación,
an additive effect of bringing the cystine much
la
lower again.
combinación es la que lograba los niveles más bajos de
We looked at
cistina.
other tissues such as the heart,
Y
liver,
nos
and
fijamos
lung,
también en
spleen, brain, and
otros
muscle.
tejidos
And you
como
can see here the heart, liver, spleen, and
riñón,
brain all had better reaction or
corazón
better
y
depletion of
los
cystine from
pulmones. Y
their
vemos que
combination treatment
también
instead of cysteamine on its own.
la combinación era mejor
And
para
in the
los
muscle
resultados de la
then, we
cisteamina.
did see an increase, but there was a lot of
Es
variability
verdad
in
que
these samples,
los
and we're actually
músculos
re-looking at those now.
había un pequeño aumento.
In terms of urine output and
En cuanto
water
a
intake,
la prurigia y la
again, here
pruriuria,
is the vehicle group in wine.
el vehículo,
Blue is
el tipo de
everolimus
vehículo es el
and
que está en color
cysteamine in red,
vino,
and then the combination
y
you can
la
see here
combinación
is much lower than
de
the vehicle
nuevo
and
nos da
everolimus groups,
los
and lower again
niveles
than
más
cysteamine.
bajos.
And again, for urine output, there's again a nice
En cuanto a
reduction in
producción
the
de
output of
orina,
urine following this 6 months
a
treatment.
través de los 6 meses vemos cómo también cae en aquellos
We looked at
ratas
Fanconi
tratadas
syndrome markers
con
such as total
combinación.
protein,
En síndrome
and
de
we saw
Fanconi,
again that
proteína
the
total,
combination treatment significantly
vemos como el
reduced
tratamiento
the amount of
combinado
protein in the urine
reduce
compared to
muchísimo
cysteamine on its
la
own,
cantidad
and also
de
glucose and
cistina en la
calcium.
orina
And just when you look at the trend of this increase, so in here, this is on no treatment, and you can see the steady increase trajectory of the Fanconi syndrome over the life course of the rats. whereas with our combination treatment, this is much more reduced and leveling out.
Esta línea es más sostenida, es más reducida.
When we looked at the histology of the kidneys, we can see here compared to the wild type, again, the vehicle, large and pale kidney. Cysteamine was able to bring back colour. Everolimus didn't do much on its own, but the combination brought it back to a much more wild type looking kidney.
Tiene ese color, pero si vemos el mejor color, lo conseguimos con la combinación de tratamientos.
And then we looked at a histological level, so this is looking at when you do slices of the kidney and looked at under the microscope.
En el riñón,
and you can see here, this is wild type, so you get this nice or histology of the kidney. So this is the glomerulus here, and these are proximal tubules, and they're meant to be nice and full and not much white gaps. You can see here in the vehicle animals, the cystotic animals, a lot of this white space, that's dilated tubule, a lot of degeneration and loss of cells. Cysteamine was able to reduce this amount of white space as was everolimus a little bit. However, the combination therapy, you can see compared to the mono treatments, had a better effect on the histology. And then we did this histological scoring, so this is just taking 10 pictures of each section, and then we do these counts, and we have this criteria. So we're looking at this proximal tubule atrophy, which is this disintegration of the proximal tubule, and then dilation and effacement, just losing these cells. And we give the score, so depending on the number of tubules that are affected, they get a percentage score, and then you get a color code. So red's obviously really bad, orange is moderate, mild, and then green is none. And as you can see here, compared to vehicle, which is untreated animals at 6 months, with the combination, we were getting much better reduction in basement membrane thickening, tubule dilation and effacement, and a better effect on proximal tubule atrophy compared to the mono treatments. And again, I'd like to reiterate that this dose was low compared to what you could give in clinical setting. So to summarize, the combination treatment versus mono treatments, in all our parameters of urine output, water intake, Fanconi syndrome markers, cystine depletion, and kidney preservation, they all did better with combination treatment. I just want to note that there were some unwanted side effects of everolimus, which are known in the field. So everolimus can cause weight loss, which it did in our rats, and we've since looked at a lower dose of everolimus and repeated this study and shown that it can be mitigated, so the weight loss has gone away, but we still get the same positive effects on our parameters here. To conclude, I just like to say combination treatment again, compared to everolimus and cysteamine, over 6 months treatment showed better reduction in cystine levels, enhanced improvement in Fanconi syndrome markers, and kidney appearance was better compared to treatment alone. And we're also completing studies with the combination of CF10 and everolimus, which are ongoing. We're going to start some new ones in Cork, but in Auckland, we're doing these studies, and the first round of results will be in, I think, September. And then I think that these new treatments have the potential to improve lives of cystic patients, and that one day we can get to the clinical stage. And just like to thank the lab in Auckland, so Professor Alan Davidson, who's my supervisor, Pang Yok Chung, who's a PhD student, Jawa, who's our technician, and Robert now has also joined the team, and also our collaborators and our funders, of course. And yeah, thanks very much.
I'm going to invite Herbie back.
Vamos a invitar a Herbie al escenario, al orador anterior.
Okay. And we'll, so if Herbie joins Jen, that was a fantastic talk, and the whole session has given us a major hope and possibility. But I think Will wants to.
It's just a quick housekeeping thing. Parents who are going to take their children to the Science Museum, which is leaving now, so if you need to go down to collect your children from childcare, now's the time to do it. So, okay. Thank you.
Thank you, Will. So we'll open the session up now to any questions. We've got some roving mics available. Please feel free. There's 2 hands up over here at the back, one in the middle. So if you're happy to, just say who you are and where you're from, and we look forward to your question.
Hi, I'm Lauren, and I'm from the UK. I have cystinosis. My question is, the
Mi
first
pregunta
clinical
es, el primer
trial
estudio clínico
for the
para el
prodrug
profármaco
in
en
Birmingham, is there a
Birmingham,
reason why they're all
¿por qué
male?
son todos hombres las personas que forman
That's an
parte
extremely
de
good
este estudio?
question.
Es una
So
excelente
we want to do
pregunta.
this as
Lo
quickly
queríamos
as
hacer
possible.
pronto, ¿de acuerdo?
And whenever
Rápido.
you
Y
treat
siempre que
a
hay
woman
una
of
mujer
childbearing
que
age,
está
there
en
is
edad de
always the
procrear,
possibility,
está en
and I
edad
appreciate in
gestante,
cystinosis this is a separate and
siempre
important topic,
existe
but
la
there is always
posibilidad, y este es un tema también
possibility
importante en
that
la
there could
cistinosis,
be a
de que se queden
pregnancy that is
embarazadas y
undiagnosed
que
or
no se
not diagnosed,
sepa al
unknown about at
principio,
that
¿no?
time.
Que no se sepa
And so it's
y
really
hay que
just to
esperar.
do this as quickly
Así
as
que,
possible.
como
And a
queríamos
similar
hacerlo
answer
rápido,
around children.
pues lo
Why
hicimos
aren't
así.
these first
Lo
three
mismo con
patients
los
potentially being a child?
niños, ¿no?
Exactly the same explanation. With a child, understandably and correctly, there are a number of
niños,
additional
un paciente
hurdles you
pediátrico,
have to
hay que
get
pasar también
over
por otras
before you can
pruebas
offer a child
antes
an experimental
que
medicine.
incluir
So we're going for the quickest and
niños en pruebas. Por
simplest
eso
way
siempre
of
intentamos
getting to this
ahora
critical proof of principle answer
ir lo más
that will allow
rápido
us then
posible.
to get into studies which absolutely will be
Después,
both
por
male
supuesto,
and
cuando
female
tengamos
to
los
start with,
primeros resultados,
adults to
por
start with,
supuesto que vamos
but
a
the
incluir
moment
a
we're confident
mujeres.
things are looking good,
Pero
we'll
primero
write that
queremos ir
pediatric
rápido,
protocol and get going on that
tener
as well.
también, por
Thank you.
ejemplo, las recomendaciones pediátricas.
Hi,
y
I'm
después
Avril
pasar
from
a
the
ello.
UK.
Soy
My
del
question
Reino
was
Unido,
very
mi pregunta era
similar,
muy
but
parecida
just
a la
regarding that,
anterior.
how long
ya
do
que
you
tengo el
anticipate the male trials to
¿Cuánto cree que
last,
van a
and
durar
when would you be
estos
looking at like
clínicos
further
con
volunteers
hombres
to be
y
involved
cuándo
in the
van a pedir
trials
más
after that? How
voluntarios
would one get
de
involved?
todo género?
Another brilliant
Otra
question.
pregunta
So,
fantástica.
because it's a
Como
single
es una
dose,
dosis
we hope
única,
we'll have the answer to this initial 3-patient
tendremos,
study by Easter next year, which will
en
allow us to
Semana
get in the
Santa del año que viene tendremos ya los primeros
application into the
resultados. Eso
MRC.
nos
We're
permite
going to make
hacer
enough
una
drug in this
solicitud al
running
MRC
3-patient study to start
y
the phase 1.
empezaremos
So we're
justo después la
hoping
fase 1.
that early
Así que
2026 we'll be in a
esperamos que a
position, if we can
principios
get
de
all the paperwork done and all the
2026
permissions in
tendremos
place
todo el
to actually
papeleo
start the phase
y
1 trial,
toda
which will
la
be
burocracia
both men and women.
necesaria para empezar la fase 1, el ensayo
Thank you,
clínico de
Herbie.
fase 1, que incluirá hombres y mujeres.
So, guys, thank you so much for just
Muchísimas
incredible
gracias por estas
presentations. I heard
presentaciones
both of these yesterday
de alta
and kind of formulated
calidad.
some questions that were in my head, but it seems like
Solo quiero
both
decir
of
que
you
ambos
are kind of attacking the same
están
question of how
lidiando con
can
el
we
mismo tema.
reduce the toxicity of
¿Cómo
cysteamine,
podemos
reduce the side effects of
reducir
cysteamine? And so
los
there's a lot of
efectos
through
secundarios
lines to
de la
both of
cisteamina?
these pieces of research. I'm
Me
wondering,
pregunto
is there any
si
benefit
hay
or have we thought
o si
about
se ha pensado
actually
en,
as
por
CF10
ejemplo, este
right,
fármaco
moves forward, is there any benefit
CF10, el
to looking
profármaco,
at
si
everomyces along
se
with
podría
it?
utilizar
Right?
con
So combining together,
everolimus,
I'm
es
wondering
decir,
if
la
that's
terapia
something that
combinada
the
del
next steps
profármaco
might be looking at as
con
well.
otros.
Yeah,
con
so we're
everolimus.
doing the
Sí,
animal
de
studies
hecho,
at the moment with
estamos
CF10 and
haciendo ya un
everolimus.
estudio
And I
con
just want to say we
animales
also
de
do our studies in male and female
CF10
rats. I just showed
y
male data just for the
everolimus,
interest of
y
time.
con ratas
but we
macho
do
y
female
hembra, por
stuff.
cierto.
But the future plan is obviously once CF10 is
es
up
que
and
cuando
running, then
FCD
it'll be starting
ya
everolimus
esté
because we'll have done the trial
funcionando
hopefully
y
with
esté todo
cysteamine
aprobado,
and
que hagamos
everolimus.
pruebas con
And it'll all
la
be there already.
combinación terapia combinada con everolimus.
Was your question about the clinical studies in man? Is that what
Pero
you
la
were
pregunta
saying?
era más
The joint everolimus and
sobre
CF10
estudios
in
clínicos en
people?
personas.
Yeah. So, so as
Como acaba de
Jen
decir
says, we will
Jennifer,
hopefully be teed up with all the
esperamos primero
preclinical data.
tener
There's a
finalizados
catch-22
los
here,
datos
and that is
clínicos. Aquí
regulatory
hay un pez que se muerde
authorities
la
are
cola,
really
que son
uncomfortable about
las
approving a
instituciones
combination study
regulatorias
with two
que no son muy
unregistered drugs
reacias
for the
a
indication.
aprobar o a ser
Obviously,
abiertas a un
everolimus is used
tratamiento,
in
aunque
the
sea de estudio,
transplant-related
con dos
setting
fármacos que
in
no han sido
cystinosis,
aprobados.
but as a therapeutic, it doesn't, to my knowledge, have a license. I'm hopeful that with your support as a community and with the solid data that Jen will generate, we might twist some arms on that one and think out the box. But I've got to manage expectations here. It may be that possibly it would need to be cysteamine and everolimus in the first instance.
Seguramente en un principio tengamos que ir con cisteamina y everolimus.
Sorry, question for Herbie. The cost of bringing a new drug
El
to
coste
market
de
can
traer
be
al mercado
expensive
un nuevo fármaco
and
es muy
can be
alto.
prohibitive when it comes to
Incluso
selling
puede
that
hacer que
drug
el fármaco
to
sea
health boards,
prohibitivo
such as
para las
drugs that we
personas
find
cuando
at
lo
the
quieran
minute.
comprar.
Can you talk around what's being
¿Qué
done with
van a hacer al
CF10
respecto
to
con
make it
CF10
more
para que no sea tan
available?
caro?
So, brilliant question,
Gran
Roy. Thank you so
pregunta.
much. And one that we are
Y una
particularly aware is
de
of
la que
considerable
somos
interest of the community,
muy,
given
muy
the scars that you carry
conscientes.
from processing.
Y sabemos lo que pasó
So
con
Ros was
Procisme.
always absolutely adamant
Ros
that
siempre
CF10
dijo
should get to
que
every
el
patient that
fármaco
needs it and
CF10
that cost
tenía
should
que
not
llegar
be a
a
barrier.
todo el mundo y que el
When you
coste
apply
no
to the
tendría
Medical
que ser
Research
un
Council
problema.
for
Cuando
funding,
pedimos
it is
y se pide
mandatory to have a
financiación,
route to
es
market
obligatorio
mapped
tener un
out in
mapeamiento
your
de cómo llegar al
application
mercado. Es una
because
solicitud que se hace al
the Medical
comité,
Research
porque
Council
el
get their government
Comité
money
Research
to improve
Council
the health and the
tienen
wealth
que
of the
mejorar
nation
la
is the
salud
mantra.
y también
What we've
el
done
estado
is set up a
económico.
spin-out company called
Hemos creado
Sundara
una empresa
because that
spin-off,
allows us to
Sundara,
maintain control over
porque
that route.
solo
And
así
Samat
vamos a
Haslak,
poder
who's the
tener
chief
cierto
exec of that
control sobre
company,
los
was
precios.
here earlier on. Unfortunately, had to go this
Había
afternoon.
alguien de
Sama,
aquí, de esta
likewise,
empresa.
is not going to see price as a
Y
barrier
lo
to
hemos hecho
access for
para que el
patients with
precio no
cystinosis.
sea
So
una
please,
barrera.
please, please rest assured
Pueden
on
estar
that point.
tranquilos
Very
al
important, and we're
respecto,
absolutely wedded
porque
to this
sabemos
being
lo
a cheap and
importante
affordable
que es
medicine.
y lo tenemos claro y lo vamos a conseguir.
Thank you.
Gracias.
Are there any other questions?
¿Hay más preguntas?
So I had a couple of questions for
Tengo
Jennifer.
algunas preguntas para Jennifer.
First of all, if now a patient with
Si ahora
cystinosis were
un
to get a
paciente con
transplant, do you
cistinosis
think there's enough data to
recibe
recommend
un
everolimus as a first line of
trasplante,
treatment?
¿recomendarías everolimus como primera línea de
I would
tratamiento?
think so, but
Yo
I'm
creo
not
que
the
sí, pero yo no
doctors.
soy, al final no soy la doctora, no soy la médica.
So can I just push you a little bit further on that line? Because is there a natural experiment
Hay
here
un
where
experimento
we knew
natural
everolimus or the sister drug
que sabemos
sirolimus
lo que
are
hace los resultados
used
de
in, not all that
everolimus
often, but in
se ha
transplantation
usado para
already.
algunos trasplantes.
Are the patients with, do we know of patients with cystinosis
También
who
sabemos
are
de
taking cysteamine
pacientes con
and have chosen or
cistinosis
been
que
chosen for
toman
their immunosuppression?
I'm taking
cisteamina y
everolimus,
toman
So
también
here we go.
but for immune
everolimus.
suppression.
Yo tomo
But it was not the
everolimus
first line of
para
treatment, so I actually started
el
with
sistema inmunológico, pero
Mifortic,
no fue mi línea primera
which is
tratamiento.
mycophenolate, but I switched to everolimus
Pero
for different
cambié.
reasons, not because of your research.
Primero porque los datos de la
About
investigación lo avalan.
3 years
Hace
ago.
10
So that
años
was actually my
que
second
empecé a tomar
question. If you
everolimus.
have been able to look at data of
La segunda
patients who,
pregunta
because of
es,
transplant,
¿hay datos
are taking
sobre
everolimus
pacientes
and
que
it
tras un trasplante
might be
toman
interesting to look
everolimus?
at?
Porque
No, I
creo que
think the
es
data has to be out there.
información
There has to be like there's patients
importante.
with
Esos
transplants and
pacientes
they have to have
con trasplantes que han recibido
the
trasplantes
what they're taking.
tienen que hablar
And I have
del
tried to
tratamiento
get
que
this
reciben.
information, but it's tricky. But I know Stephen. Is he
¿Steven
here?
está aquí todavía?
Did you look in the American
Porque creo
side?
que lo ha mirado
You told
en
me.
la página de Estados Unidos. El comentario es fuera de micrófono. No nos llega el sonido a la cabina. Así que no podemos traducir las palabras de Steven.
Yeah, so if we could ideally get like a cohort of
Tendríamos
people
que
who are
tener
already on
una
these and then
corte
do some
de
measurements before and
personas
after.
que ya estén tomando
Obviously not
everolimus
before because
y
that's gone.
hacer una
But, from
comparación
now on and see what the progression
antes
is.
y
But
después para ver cuál es la
So I think we
progresión.
could develop that actually. We can go to the
Podríamos, de
European
hecho,
NHS Blood and Transplant
hablar
and to
con
the
la
scientific
Asociación
registry
Europea
in the
de
States maybe, just to
trasplantes
see
y
if
ver
there
qué
are,
comparativa
if
hay
almost in a way, if that
con
natural
los Estados
experiment has
Unidos
been performed. So
y ver cómo los experimentos
I
naturales
think
se han
that's
llevado
very
a cabo.
exciting. We'll be in
Y
touch.
es muy, muy,
And
muy
then a third question
interesante.
that's more of curiosity. If
Tengo una curiosidad.
if you have an estimate if the dose
Han
for this
calculado
case,
si
not
las
for
dosis para
immunosuppression,
estos
how
casos
it would
no,
compare to the one used for
no las de
immunosuppression for people
inmunosupresión,
with
cómo se
transplants? If it
comparan
would be the
con
same
las
dose or
dosis
lower? Because
de
of course we know that
inmunosupresión,
this treatment also has
porque
other
sabemos que
side
tiene otros
effects.
efectos
exactly,
secundarios.
yeah. And so originally we did choose the high dose of 10 mg per
Podemos
dose.
escoger la dosis de
And
10
we've gone right down now
miligramos
to 2
por
milligrams, in our new
kilo,
studies.
pero lo
So
hemos bajado a 2.
ideally, we'd wanna
La
go
idea
low,
es
but-
bajar al
and it
mínimo.
wouldn't be for
No
immunosuppression,
sería
this
para
would
la
be for,
inmunosupresión, sino para los
combination
tratamientos
treatments, so it'd be a different
combinatorios.
dose what you'd
Sería
get for
una dosis
immunosuppression.
distinta.
So, yeah, I don't know
Así
how
que
it would
todavía
work
no
if
sé
you're already on it for
qué
immunosuppression
pasaría
and then
si
taking the
primero
combination.
hubieran
Yeah.
estado tomándolo para el trasplante y
So
después
even at 2
con
milligrams
combinación.
per
No,
kilo,
mostly-
it's
2
still a lot
miligramos
more
ya es
than
Mostly
mucho
in the case
más.
that someone who doesn't need the immune suppression because they don't have a transplant. If
Si lo
they take
toman
it
no para la inmunosupresión, sino para la cistinosis, sería interesante si se tomarían la misma dosis que para la inmunosupresión. No, imagino que sería muy inferior la dosis. Creo que esto abre una oportunidad muy interesante. Los estudios clínicos para ver cómo podría funcionar son factibles, yo creo, en una escala temporal más o menos razonable. Así que vamos avanzando tan deprisa como podemos, desde luego. Gracias. Quizás no puedan responderme pero ¿cuáles son los calendarios, más o menos, respecto al CF10 y los ensayos clínicos que están a punto ya de empezar a estar disponibles para de aquí? ¿Cuánto tardaremos en que esté útil en clínica, que esté usable en clínica? Nosotros pensamos empezar los estudios clínicos de fase 1 en 2026. Y ya sé que esto parece una eternidad, parecen tiempos arqueológicos, pero el equipo de Sunderland y hay un ejército de gente que está trabajando en esto y vamos tan deprisa como podemos. Pero la respuesta a su pregunta dependerá también de las autoridades regulatorias y de lo que nos pedirán que proporcionemos para poder registrar el fármaco. Todavía no hemos empezado esa conversación. Lo que sí sabemos, y de eso hablaremos el primer día, es qué vamos a hacer con los pacientes que están como voluntariamente en el paciente de manera altruista, empiezan a tomarse el fármaco durante 28 días, funciona bien, sin efectos secundarios, como esperamos que pase. ¿Qué le vamos a decir a esos pacientes cuando lleguen al día 29? Mire, lo siento mucho, tiene que volver usted al sistema de mierda de antes de la cistemina. Y los pacientes dirán, no, oiga, yo quiero seguir tomando CF10, así que eso lo vamos a preguntar directamente a la administración, porque son datos que podrían convertirse en un paquete de registro, así se llama. Es un debate que vamos a tener con la administración, pero bueno, lo que buscamos es la vía más rápida para poder registrar este fármaco, para permitir que el CF10 esté disponible de manera mucho más generalizada de lo que estaría al principio. Gracias. Aquí hay otra pregunta. Es un comentario respecto a la combinación de cistemina y everolimus. Como decía Steven, el everolimus casi no se usa en trasplantes. De hecho, en Reino Unido no está disponible para trasplantes, así que después de un trasplante, además, ya no tienes síndrome de Fanconi porque tienes un trasplante y eso es lo que mejora mucho, sobre todo en el Fanconi. Así que, ¿cómo serían las lecturas si se combinan cistemina y everolimus? No sé si será más fácil conseguir una liberación de los sistemas de los niveles de cistina. No sé. Ayer y hoy hemos hablado de ese tema. Quizás sería para personas con síndrome de Fanconi que todavía no tengan trasplante de riñón, seguramente esa sería la necesidad no cubierta a esas personas con síndrome de Fanconi. Y si les das Cistagón más everolimus o Procisbi más everolimus, quizás ese sea el grupo diana más importante. Sí, estoy de acuerdo. ¿Más preguntas? Con eso hemos llegado al final del tiempo que teníamos asignado, así que un gran aplauso y un gran agradecimiento para Harvey y Jennifer.
Real excitement ahead. The next exciting development, I was just saying, running with Neil, the adult
se refieren al grupo de cistinosis de los adultos. Uno de los temas que hablábamos eran los colirios y las complicaciones oculares oftalmológicas. Aquí también hay una necesidad sin cubrir. Se esperan más avances en este campo y es un placer presentar al profesor Anuj Chawan, que es catedrático de la Escuela de Minería de Colorado y está trabajando en tecnologías que también pueden llegar a los pacientes con cistinosis y pueden ser útiles para ellos. Bienvenido. Muchísimas gracias por esta oportunidad de intervenir aquí y con muchas ganas de escuchar todas las preguntas, porque todo esto lo estamos haciendo para que estos productos lleguen a manos de todos ustedes, así que su participación será muy importante. El título es un poco complejo. A ver, ¿dónde tengo el micrófono? Que no me lo deje.
Yeah, the title is a bit complex, but really what we're trying to do is to replace the eye drops with
sustituir el colirio con lentillas, con lentes de contacto. Les explicaré por qué queremos hacerlo y por qué va a funcionar.
So all of you, I think, are familiar with some of the
No sé si han
challenges
visto
of eye
ustedes
drops.
los problemas que representan
The
los
systemic eye drops that you take right
colirios,
now, it could be either
que
Cysta
pueden ser
drops
o
or
el
Cysta
Cysta
Gone.
drops o el Cysta Gone.
It has
y
poor
tiene
bioavailability.
poca
What it means is that
biodisponibilidad. ¿Qué significa eso?
when you
Pues que
put a
cuando
drop
ponemos una gotita
of drug into the
en
tears,
la
the
lágrima,
drug goes
eso va
everywhere
por
in the
todo el
eye.
ojo.
Now,
Pero
what
lo
you
que
want
queremos
is, you
es
want the
que
drug to
el
go
fármaco
into the
entre
cornea.
en
So
la
this is the structure of
córnea.
an
Esta es la
eye,
estructura del
where
ojo. Esa parte en
the
amarillo
yellow
es
is
la
the
conjuntiva.
conjunctiva. This blue is the contact lens. I'll
azul
talk about
oscuro
that
es
in a
la
minute.
lente de
And this
contacto y
gray
el
is the
gris la
cornea,
córnea, la
the
parte
center
central.
part.
Y lo que
So you
queremos
want
es
the
que el
drug to go
fármaco
from the
llegue desde la
tears
lágrima
into the
hasta la
cornea,
córnea,
because
porque
that's
ahí es
where all
donde
the
están los
crystals are.
cristales. ¿Qué
What will
ocurre?
happen is the
Que
drug
el
is not
fármaco
going
no
to just
solo se
stay
queda aquí
here. When
cuando
you
la
put it
ponemos
in
en
the
el
eye,
ojo,
it's
no,
going to go
sino que
everywhere. All of
se
these tears,
desparrama por todas
they're going to have your
partes.
drug.
Toda la lágrima
And a lot
tendrá
of the drug will
fármaco.
go
Y una
to
parte
this
muy grande
yellow
del
tissue
fármaco acabará
into the
yendo
blood.
a la conjuntiva, a la parte en
And
amarillo,
also what
y de
happens is
ahí
every time you blink, you
a la
actually
sangre.
push
Y
some of the
además,
tears into your nose.
cada vez que
There are these two
parpadeamos,
holes in the
parte
eyes
de la
called
lágrima
puncta,
va
which
a la
connect your
nariz.
eyes to your
Tenemos estos dos
nose.
agujeritos
So what
que
happens is
conectan
most of the
el
drug
ojo
that
con la
you're putting
nariz,
into
y
the
gran
eye
parte
doesn't actually go into the
del
cornea.
fármaco que ponemos
It
pues
goes to the
acaban
rest of
en
the
otras
body.
partes del cuerpo.
In
El
fact, about
95,
95 to
entre el
99%
95 y el
doesn't go.
99% no llega
And
donde
so
tiene
that
que
number
llegar.
is
Así
really
que,
low, and the
bueno,
question is, can we
eso lo
improve
podemos
that?
mejorar porque es poquísimo.
But because that number
Al
is
ser
very
tan
low,
bajo,
you have to put
pues
a
hay
lot
que
of
poner
drops. You have to
muchas,
put 8 drops or 4
muchas
drops, depending
gotas,
upon your
8
formulation is. So if you can increase this number,
gotas,
you will
por
not have to
ejemplo,
put that many
o
drops.
10. Y si
Another
mejora la
challenge is that the
biodisponibilidad, no
drug
habría que
cysteamine,
poner
it can
tantas.
very easily
Además,
oxidize
la
into cysteamine.
cisteamina se oxida
And that's why your
rápidamente en
formulation comes
cisteína,
frozen,
entonces
so that the
aquí la
oxidation
oxidación
doesn't
ya no
happen.
se produce.
Sorry. There's also stinging
Además,
with the
también
eye drops.
producen
And one of the reasons for the
picor.
stinging is
¿Por qué?
the drug itself could cause stinging.
El fármaco no
But in addition to
provoca
that, there is a preservative in there
picor,
called
pero hay
benzalkonium chloride.
una
Because
conservante
you don't want the drug
sí lo
formulation to
provoca.
develop
No
have
queremos
bugs
que
going in
los fármacos tengan
and to
bichitos,
prevent bugs
¿no?
from growing in it.
Para
Because
evitar
the air is
que
full of
crezcan
bugs.
los bichitos que
So
hay en el
when you-
aire y en
when you
todas
open an eye drop
partes,
bottle, some
cuando
bugs
abrimos
could
la
enter the
botella
bottle,
de fármaco,
and then if
pues
there's nothing in there to
entran
kill them, they will
bichitos
grow.
del
And
aire.
you don't
Y
want
si no hay
that,
nada,
so you put
si no
preservative
ponemos
in there.
conservante,
And when you're putting an eye drop
pues
into your eye, the
crecerían.
preservative
Y
is going
entonces,
into
¿qué
the eye.
pasa? Pues que hay
And you're doing
conservante
this
y
8
estamos
times a day.
poniendo
And so
también
there's a
mucho
lot of
conservante
preservative
en
that's
el
going
ojo
into your
porque
eye
nos ponemos
that can
8
cause
veces al
stinging.
día
It can
la
also actually
hoja
damage
y
your
eso también
ocular
puede
epithelium. So you don't want
dañarnos
that,
el ojo.
if possible.
Si fuera posible, pues mejor
And finally,
no
the bottles
ponerlo.
have
Y
to be
además,
shipped frozen to
las
minimize the
botellas hay que
degradation. So there are all
enviarlas
these
congeladas
challenges,
para minimizar
and
la
depending upon what
degradación
formulation
del
you use, there are
fármaco.
additional
Además,
challenges, like the cysta
depende
drops are very
del
viscous,
fármaco, pues
they're
algunas
difficult
son
to put
viscosas,
in,
difíciles de
they
poner,
could blur your
o
vision.
pueden,
So there are a lot of reasons why
hacer
we
más borrosa la
might want to
visión. Así
do
que hay
all the
muchas
drops
cosas
and probably a
que
better
nos
approach.
que
So if you
podemos
think
mejorar.
about what could be a
¿Y
better
cómo
approach,
podríamos
what
mejorarlo
we're
todo
doing is
esto?
we want the
Queremos
drug
que
to go into the eye.
el fármaco llegue al
That's
ojo,
the
¿no?
requirement. Eye drops you have to put
Las
again and again
gotas
and
hay
again
que
and again.
ponerlas una
Wouldn't
y
it be
otra vez.
nice
Y
if
no
you put
sería
something one
fantástico poner
time
una cosa una
and that
vez
slowly
y
keeps
que poco a
releasing the
poco
drug?
fuera liberando el fármaco?
So
Esa
that was
era
the
la
basic
idea
idea
que
of
inspiró
starting
este
this project.
proyecto.
And the question is, okay, what do
¿Y
you
qué
put
se
into the
pone en el
eye?
ojo?
You could put that product
Bueno,
that you
pues
want
un
to release
producto
slowly
que
at many
podamos
locations. You could put that in the injectable sac. But a natural
que
device in the
podamos- que se
eye
libere,
that hundreds of millions of
liberar
people in the
lentamente,
world are
pues
using are
es
contact lenses.
fácil en otras partes del
So we
cuerpo,
thought
pero
instead of
pensamos
creating
que
a new
ya
device
que hay
that you
tanta
put
gente
somewhere,
llevando
why not just
lentillas,
use
¿por qué no
contact
usamos
lenses
lentillas
that
que
are
ya se
already being
usan,
used, that have
que
been
son
proven to be
seguras, se ha
safe?
demostrado,
Why
que
don't we
mucha
put the
gente
drug
ya las
into
usa?
that
¿Por qué no
contact
ponemos el
lens
fármaco
and then
dentro
use
de la
that as a
lentilla y ese
delivery
es el
vehicle?
vehículo
So that was the initial
para
idea
liberar el
that
fármaco?
we
Esa es la
had
primera
when we
idea
heard
que
about
se nos
this
ocurrió
problem.
cuando nos enteramos de
Today I
ese
will try to show
problema. Y
you
hoy
we have come
hemos
quite
avanzado ya
far
bastante
in actually
para
translating that
convertir
into
esto
actual
en
practice.
una realidad.
So when
Cuando
that
se
idea
nos
came to us,
planteó
we
la
thought, okay,
idea,
let's just take
pensamos, vale,
a
muy
contact
bien,
lens,
tomemos
soak
una
it in your drug
lentilla,
solution,
vamos a
so the drug will
poner
go into
dentro
the
de la
lens,
lentilla
and
la
then let's
solución
measure at what rate
farmacológica
the
y
drug
vamos a ver
comes
a qué
out.
velocidad sale
So
el
when we made that
fármaco.
measurement, what
Y
we saw
vimos
that
que
the percentage
el
releases, what percentage of the drug is
de
coming out as a
fármaco
function
que
of
se va
time.
liberando en
So what
función del
happened is
tiempo,
that
pues
the
bueno,
contact
las
lenses that we
lentes de
tried,
contacto
the drug came out very quickly, in about
sacaban
0.2 hours, which is
el
basically 10
material
minutes. In
salía
10
al cabo
minutes,
de
the drug
muy
is
poco
all out.
tiempo, en 0,2
So that was
horas,
never going to
0,4.
work,
Esto no va a
because if
funcionar
in
porque
10
al cabo de 10
minutes, that
minutos
would
ya
mean
está. Quiere
that you
decir
load the
que
contact lens with drug, you put it in the eye.
al
After
cabo
10
de 10
minutes, all the
minutos
drug
ya
is
se ha
gone.
ido todo
So
el
then you'll
fármaco,
have to
tendrás
put
que
it onto
ponerte
the
otra
lens.
lentilla nueva.
And the
Y
only
lo
thing worse
único
than putting 8 drops would be to put 8 contact
peor a
lenses. So
ponerse
that was never
gotas
going to
8
work.
veces al día será ponerse
So we said,
lentillas
okay,
8 veces
so
al
our
día.
first
Entonces,
challenge
primero
is to
vamos a
design a
diseñar
contact
una
lens that will release the
lentilla
drug
que
slowly.
libere el fármaco lentamente.
So to do
Para
that, we
eso
came up with a very
se nos
simple
ocurrió una idea
idea.
muy sencilla.
The idea is
Y la idea
this.
es
Imagine
esta.
that
Imaginémonos
this is
que
your
esto
contact
sea la
lens.
lente de contacto, la
And
lentilla.
these are your drug
Y
molecules.
queremos poner aquí las moléculas.
So the molecules,
Las
basically what
moléculas del
happens is when
fármaco.
you put the
Cuando
lens, the
ponemos
drug
las
molecules just get out of
lentillas,
the lens and
pues
go into the
esas
tears.
moléculas acaban pasando a la
Then the
lente.
idea we
Y
came up
la
with is, what
idea
if you put
es,
barriers in
¿y
the
si
path of
ponemos
the
una
drug?
serie de barreras,
imagine they are bricks.
barreras como si fueran
And the drug cannot go
unos
through the bricks, the
ladrillos,
drug has
tienen
to
que
go around the
rodearlos
bricks.
porque no pueden
So with that
cruzarlos.
idea,
Así
we
que
tried a few
probamos
different things, and we were able to
distintas
successfully
cosas y
create those
conseguimos
barriers in the
crear unas
contact
barreras
lens.
dentro de la
So
lentilla.
this was the
Esto es el
schematic
esquema de
of
lo
what
que
we wanted to
queríamos
do.
hacer.
This is
Eso
what
es
we
lo que
actually
llegamos a
did.
hacer.
This is the
Esta
contact
es la
lens
matriz de la
matrix.
lente de
The
contacto.
picture is a
Esta
very
es
high
una imagen
magnification
muy
picture called
ampliada.
SEM.
Y estos
And these bricks that you see, they are actually made up of vitamin E, which
ladrillitos
is very, very
son
biocompatible.
de
It's actually an
un
antioxidant. It's
antioxidante
actually good for your
muy
eyes.
biocompatible que se
So
pone
that
para
was the
los
idea,
ojos y la
create these
idea
bricks of
era
vitamin E,
poner
so the
estos
drug will have to
bloques
go
de
around them,
vitamina E
and that will
que
hopefully lead to
obligaban
longer
a las
release.
partículas, a nuestras moléculas, a tener que rodearlas y
Okay,
esperábamos
let's
que
see.
tardaran
So yeah,
un
that's
rato
the idea.
más,
The drug will
¿no?
have to go
En
around
salir.
them. And this is just how we make the barrier, so it's
Y
not
bueno,
really that
esta es la
important
manera de
stuff.
fabricar la barrera.
So I'm
Así
just going
que
to show you the proof that indeed it
vamos a
works.
demostrar que esto sí
So
funciona.
this is
Estos
the
son los
data.
datos. Antes
I already
hemos
showed you the
visto
data at the
arriba
top,
la gráfica de
where
arriba,
all
que,
the drug
en que
comes in
todo
about
el
10
fármaco
minutes. But
sale en
now,
10 minutos,
after
pero
putting
después se ha
vitamin E
puesto
into the
vitamina
lens,
E como
now
barrera
here
dentro de la
we're looking
lente.
at
Miren,
AQV Oasis
estas son
lenses, which
unas
are one of the most
lentillas
comfortable, one of the
AQV
most popular contact lenses in the
Oasis, que
world.
son de
Like I said, with hundreds
las
of millions of patients
más
already
cómodas
wearing
del
it.
mundo. Hay millones de
And what we
pacientes
have
que
is the
las llevan, que las
percentage
llevan.
release, that
Y
is how
vemos
much
el
drug is
porcentaje de
released over
liberación de
time.
fármaco
And you
en
can
el
see
tiempo.
the
Abajo
one without
tenemos
vitamin E,
las
everything
horas.
comes
Y
out
vemos
like
que
literally in a burst.
si no
But with
ponemos
vitamin
vitamina
E,
E
you can
sale todo
stretch it
junto, pero
to whatever
si
time you
ponemos
want,
vitamina E,
depending
en
upon how
función de
much
cuánta
vitamin E
vitamina
you put in.
E
The more vitamin
pongamos,
you put in,
podemos alargar
the longer
el
the
tiempo
drug
de
is
liberación
released.
del fármaco.
And you
Cuanta
can
más
see
vitamina E,
with about
más
20%
tarde, con un
vitamin E in the
20%,
lens, you can
pues
release for
pueden
about
estar
4
4
hours.
horas
And
liberando
with 30%
fármaco
you can
y
go
con
to
un
about 6
30%
hours.
pues
And that's really
hasta 6 horas.
what we
Y eso
need.
es
You don't want
suficiente.
to
No
release for
queremos que se
multiple
libere el
days.
fármaco varios
Because you want a
días, porque
different
cada
lens every
uno
day.
se
You want
pone
disposable
lentillas nuevas
lenses.
cada día.
So the idea
Son de un
is
solo
you put
uso,
the
así
drug
que
in
le
the,
pones
you'll
el fármaco
load the drug
a
in the
la
lens is
lentilla
going to die. When the drug is all
y
gone,
cuando
take the lens
ya
out,
se
throw
acaba
it
el
away.
día o se ha
Next
salido,
day you'll put
pues
a new
se
lens.
quita la lentilla y al día siguiente lentilla
Okay,
nueva.
so this
Esto
this was very
parecía
promising.
muy
So
prometedor
we said,
y
okay,
dijimos, vale,
and
muy
this is
bien,
a
esta es la
picture of
imagen
what
de
a
cómo
lens
sería la
looks like.
lentilla,
It's very completely clear.
transparente,
It's
un
slightly
poquito
yellowish because
amarilla,
that's the
porque
color of
este
the
es el
vitamin.
color de la vitamina
So then we
E.
started
Así
testing
que
it
empezamos
for
a hacer las
safety.
pruebas de
As you
seguridad,
saw,
todos
any
los productos
product,
como saben,
first thing you have to
antes de
do is before you
empezar
start looking at
a ver
efficacy, you
la
have to prove the
eficacia,
product is
primero hay que
safe.
ver que sea
So
seguro.
we made
Así
these
que
lenses and we
pusimos
put them in rabbits.
estas
It
lentillas
was a
en
7-day study.
en conejos, estudios de
And
7 días.
during
y
the 7
durante
days,
esos
there was
7
no
días
toxicity.
no
I'm
presentó
not going to go into
toxicidad, no
details of all
entraré
the studies that
en
were done,
detalles de
but
los estudios,
essentially there
pero
was no
vaya resultados
toxicity.
que
And
no
so
eran tóxicos.
we
Y
said,
así que
okay,
dijimos,
let's test
vamos
if
a
the
ver si
idea
esto
works.
funciona.
Now, you might have seen that a
Habrán
lot
visto que hay muchos
animals
animales
that were
que se han
used for a
usado
lot of
para
the drug
hacer
testing are
pruebas
mice.
de fármacos y muchos son ratones.
It's
Es
challenging to put
difícil
design contact lenses
ponerles
for mice. We actually have
lentillas
design
a
contact
los
lenses for mice,
ratones.
but it's not an
No
easy
es
study to
fácil,
do.
aunque lo hayamos hecho algunas veces.
So
Así
we
que
had to come up with a
dijimos,
different
bueno,
animal
busquemos otro
model.
modelo de animal. Y
Some
algunos
researchers
están
are trying to
intentando
create a
un modelo de
cystinosis model in
cistinosis con
rabbits,
ratones,
but
pero
right
ahora
now there is
mismo hay
there's no
modelo
reliable
fiable
model.
para ello. Así que dijimos,
So we
bueno,
had to come up with a very simple way
busquemos una
of
manera
testing whether the
sencilla de
drug
ver si
works. So
esto
what we did
funciona o no.
was that we took,
Tomamos
the rabbits are frequently
a
used for
los
eye
conejos se les
studies
usa mucho por
because the
el-
size of a rabbit eye is comparable
los
to the size of a
estudios
human eye. So So we
de
took a
temas
cadaver rabbit eye.
oftalmológicos.
Cadaver rabbit eye means this rabbit was sacrificed.
Así que tomamos, porque tienen unos tamaños
food,
parecidos,
we get
conseguimos
these,
estos ojos de
we get
conejos
these
sacrificados, muchos de ellos para
and then
comérnoslos,
we
y
put
los
them in a
ponemos en una
chamber,
cámara y
and then we
después
soak it
lo
in cystine solution.
humedecemos
So cystine
con
essentially goes
solución de
into the
cisteína
eyes,
para que
and then we
entre en el ojo y
oxidize it to
después lo
convert to cystine.
oxidamos para
So we
convertir
create cystine crystals. Now,
cisteína.
I'm not going to call that
no
as a
diré que esto
cystinosis
sea un
model,
modelo
because
de
this rabbit doesn't
cistinosis,
have a
porque
disease.
este conejo no
We're just loading
tiene
it with cystine crystals.
una enfermedad, no tiene cistinosis, pero lo
And
estamos
after that,
cargando
what we
de
do is, so this is what it
cistina.
looks like.
Esto
This
es
is
una
simply a
imagen
picture, high magnification picture to
muy,
show
muy
that indeed
ampliada que
the crystals
muestra
are
que
formed, and
los
the crystals
cristales
look
se
pretty
han
much
formado
similar
y que se
to
parecen
the crystals
bastante a
in a
los que
human
se
eye.
forman en el ojo humano.
So after that,
Después
what we did was we started
empezamos a
putting our contact
ponerles,
lenses on these animals, or not the animals, but the cadaver eyes,
las
and we showed
lentillas
that the
a
crystals
los
dissolved away.
ojos de estos
So that
conejos
seemed
ya muertos
promising.
y
So what
vimos
we
que
so
se
far, based on
disolvían
the
los
data that you have
cristales, muy
seen so
prometedor.
far, it
Parece
seems
que,
that
pues,
We
se
can
puede
design a
diseñar
contact
una
lens to
lentilla
release
que libere
slowly for whatever duration we
lentamente con
want.
tanta duración como queramos
The lenses
y
are
son
safe in
seguros
animals,
en
and they
animales
also
estas
seem to have some
lentillas y
efficacy.
parecen que son
But the
eficaces. Pero
efficacy
aquí
here
la
is very qualitative. So what we
eficacia es
wanted to then
cualitativa.
see is
Queríamos
how
saber
much drug is going into
cuánto
the eye that
fármaco
will actually
entraba
dissolve the
en el ojo para
crystals.
disolver los
And that's called pharmacokinetic
cristales.
studies.
Y
Just
eso
like you
implica
saw pharmacokinetics for CF10,
estudios
we wanted to use pharmacokinetic studies. That
estudios
means
farmacodinámicos
you put
como
the
los que
lens on
hemos
the
visto
eye of
para
an
la
animal, and then you
CF10.
measure the concentration of
Ponemos la
drug
lentilla
inside
en
the
un animal,
eye,
medimos la
and also in
concentración
tears at various time
del
points.
fármaco en el ojo y también en la lágrima
So
del animal.
this is that study.
Estos son los resultados de ese
In this
estudio.
case,
La
the concentration I'm showing you is
solamente
only in
es en el
aqueous
humor
humor.
acuoso,
Aqueous
que
humor
es
is
el
that
líquido
fluid
que
in
hay
the
en el
eye,
ojo, justo
just
detrás
behind the
de la
cornea.
córnea.
And the
La
concentration
concentración
is
aparece
in
en
units
unidades
of
de
milligram per ml, or
microgramos
microgram
por
per ml.
mililitro.
So
El primer
the first
estudio se
study was
hizo
with eye
con
drops because,
con
we
el
want us
colirio.
to ourselves to be
Nosotros
at least
queríamos
delivering
entregar
drug equal to 7 to 8 times
que
the eye
el
drop. So
fármaco
we wanted
funcionara
to first
como entre
measure how
8
much
y
drug
10 veces
goes into
más
the eye of a
que
rabbit with a
con
single
los
eye drop,
colirios.
commercial eye drop. And then we'll multiply
Así que
that
si
by 8,
multiplicamos por
and that
8
will tell us how
lo
much drug
que
you want to
primero
go from
medimos
a
lo que
contact lens.
daban los colirios y
So this
después
is the
quisimos
data for an eye drop.
multiplicarlo por 8, ¿vale?
So the
Estos son
concentration
el
essentially starts
colirio,
with
empieza
0
la
just before the
concentración
eye
con
drop is
0
put
antes de
in.
poner
After the eye
el
drop is
colirio
put
y
in,
después
the
cuando se
concentration
pone
in the aqueous
empieza
humor
a
starts to go
aumentar
up.
la
It reaches a
concentración en el
maximum,
humor acuoso, llega
and
a
then
un
after
máximo y
the
al cabo
basic eye drop is
de un
completely
tiempo,
gone, the
momento
concentration starts to go
empieza
down,
a
and
bajar
eventually
la
it will go down to
concentración
0.
hasta alcanzar otra vez el
So
cero.
with that,
Después
after that, we started putting our
decidimos
contact
poner
lenses onto the eye.
la
Now,
lentilla.
to put a contact lens,
Poner
we had choice. We
lentilla,
already
ya
knew what
sabíamos
contact
qué
lens we were
lentilla
going to use,
queríamos,
but we had
pero
to pick
teníamos
how much
que
vitamin E
saber
do we want
cuánta
to load into
vitamina
the lens.
E queríamos
And that has to do with how
introducir
long
en la
do we want to
lentilla.
put the
Eso
lens on the eye.
dependía
And
de
that is something
cuánto
that actually we
rato
need your input on.
tenía
I'll come back
que
towards the end of the
llevarse
talk
la
on that.
lentilla puesta. Después podemos
So
hablar
we
de eso.
didn't
Nosotros no
want patients
queríamos
to have
que
to
el
put a contact
paciente
lens for the entire day.
llevara la
Because not
lentilla
all of you need contact lenses. Now,
todo
these
el
contact
día
lenses
entero.
will have power in them. So if you need vision
Así que,
correction,
bueno, si
then
uno
fine, you can put it for the whole
necesita
day.
corrección,
But
pues
patients that don't
fantástico
need
todo el
vision
día, pero
correction,
los que no
you have
necesitan
to ask them
corrección
to put a
de la
contact lens
vista,
for the entire
tener que
day
ponerse
just to
lentillas
deliver the
todo el
drug
día
seemed a
entero
bit too much for
para
us.
administrar
So
el
we wanted
fármaco nos
a
parecía
weird
mucho.
duration
Así
that
que
is
queríamos una duración que
maybe
fuera
just a
no sé
couple
qué
of
tan par
hours,
de hora
so that
para
you have
que
flexibility,
uno
you can
pues
do it
pueda hacerlo de
daytime,
día, de
nighttime,
noche,
whatever
sea
you want.
más
And
flexible.
so we wanted
Así
a
que
weird
queríamos
duration
una
of just a
duración
few
de unas
hours.
pocas
So that's why
horas.
we
Por
chose
eso
vitamin E of
decidimos
2 hours
vitamina
or
E
20%
20%
that releases for about 2 to 3 hours,
pues liberación
and
2-3
30% which
horas
releases about 4 to
y
6
al
hours.
30%
So
que
those
son
were the
4
two
o 6
loadings
horas
that
de
we
liberación
tested.
del fármaco. Probamos estas
So
dos
first we
opciones.
tested the
Primero
20%
probamos
vitamin
vitamina
E.
E al 20%.
And you can see with the contact
Miren
lens,
que
the
las
concentrations are
concentraciones de
much higher.
vitamina,
They
perdón,
still go down,
del
but
fármaco
even
son mucho
after
más
essentially
altas y
120
al
minutes, the
cabo
concentrations are high.
de 20
And
minutos
with
ya
a
y
single
con un
contact
único
lens,
fármaco,
from this you can
perdón, con
calculate,
una
and this contact lens
lentilla,
essentially delivers the amount of drug that is needed
pues
for about
nos
7 eye
da
drops. So we are very close, we're
la
right in the
cantidad
ballpark.
de fármaco que se necesita,
And then
que
also
se
with the 30%
obtiene con
vitamin E,
7
you can
gotas.
30% vitamin E,
Y a 30%,
because there's more vitamin E, the drug
como
comes
hay
out
más
slower.
vitamina E,
So the
pues
concentrations are
el
lower,
fármaco
but will last much longer.
sale a menor
And so in that
concentración,
case
pero
also
durante
we can
más
deliver
tiempo,
about the
así
same
que
amount
se
of
podría
drug.
administrar más o menos
But the reds,
la misma
if you
cantidad, pero si
put
pones
30% vitamin E,
E
you'll have to
al
wear the lens
30%,
for longer
la
duration,
lentilla tiene que
maybe
estar
about
más
6
tiempo
hours.
puesta,
But if you
6
put 20%
horas, por
vitamin E,
ejemplo.
you may have to wear the lens only for
En
about 2
cambio,
hours.
2 horas y
All right, so
solamente
in
de
addition to
vitamina
that,
E.
in addition to measuring the concentration
También
in the aqueous humor, we also measured in
medimos la
different
concentración
tissues.
en el humor
Cornea is where all
acuoso
the
y en
crystals
otros
are, so you want to measure
cristales, en las zonas donde
concentration.
están los
And you can see even in
los
cornea,
cristales, en
with
la
these vitamin-loaded
córnea
lenses,
vemos
we can actually get
que
higher
hay más
concentration compared to
concentración
eye
que
drops.
con el colirio.
Retina is the tissue
y
in the
en
back of the eye.
en el
There are still
tejido
some
que hay
crystals in
la
the
parte
back of the
posterior
eye,
del
so it's
ojo
still good
también
to be
hay
able to deliver
cristales,
some
queríamos
there.
que llegara ahí
And they'll also be
también
able
el
to deliver
material
at
y
pretty high concentration.
llega a buena
So that was
concentración.
good.
Eso estaba
That
muy
means
bien,
I think
quiere
we are on
decir que
track.
vamos bien.
Essentially, well, this is just a
Esos
mathematical
son los
model, so
modelos
we'll skip that.
matemáticos, nos los
So far
saltamos.
everything
De
looks
momento
good.
todo
And that
perfecto.
means the next
Significa
step,
que
typically after
el
animal studies is
siguiente
successful, is
paso
to go into
sería
humans.
pasar a seres seres
But
humanos,
before
pero antes
you
de
put the
poner este
drug
fármaco en
into the
una
humans, we
persona,
just want to
queremos
test the
probar
vitamin E.
primero qué pasa con la
Are the
vitamina
lenses with
E.
vitamin E, are they
¿Son
safe and
seguras
comfortable?
las lentillas
So we did a human
con
study with
vitamina
10
E? ¿Son
patients where we put
cómodas?
the
Así que les
lenses on the
pusimos
humans
lentillas
for
durante
2
2 días.
days.
Un día
One day they
las
wore
llevaban,
it and then
después
they
las
changed it
cambiaban y
and
se
wear
ponían
another
otra
lens.
lentilla
And
y
there was
no
absolutely no
había
toxicity.
ninguna
Patients are completely
toxicidad,
comfortable. We
totalmente
add a
cómodas.
lubricant to this
Añadimos
contact lens to make it
lubricante
very,
para
very
que fuera
comfortable.
todavía
So
más
these lenses
cómodo,
are
así que
actually
son unas
very
lentillas
comfortable,
muy,
very
muy
lubricious.
lubricadas y
So we
muy
thought, okay,
cómodas.
now we
Y
can start
dijimos,
putting
vale, pues
drug
ahora
into
ya
the
podemos empezar a
lenses.
añadir fármaco.
Now, we haven't
No
done any
habíamos hecho
clinical studies
todavía
so far with cysteamine lenses, but we've already done
con
clinical studies with other
histamina
drugs.
en lentillas, pero
So
sí
these
con otros
lenses have already
fármacos. Así
been on
que
patients,
ya
including
habían
a drug.
habían sido probadas con
So the
pacientes
drugs that we
con
have tested are
un
glaucoma
fármaco,
drugs
por
as well as
ejemplo,
anti-infective
fármacos
drugs.
para
But for
glaucoma,
cysteamine,
pero no lo
we
habíamos
haven't tested
probado
it.
con cisteamina.
Nonetheless,
Pero bueno,
we are ready
estábamos
for
ya
it.
preparados y
So
para
start doing
empezar con
human
los
studies, you have to
estudios
first
con
get what
humanos,
is called
primero se
IND
necesita
approval
la
in the
aprobación
United
IND,
States.
se llama
It's
así,
called Investigative New Drug Approval.
aprobación para
We already have received
nuevo
the IND
fármaco de
approval from the
investigación. Ya
FDA. That means
tenemos
the FDA has
la
told
aprobación
us
de la FDA americana,
we believe that your
que
product is safe
significa que nos han
and
dicho
it
que
is
creen que nuestro
fully
producto es
validated in
seguro y
terms
que
of
está válido en
manufacturing.
cuanto a la
You
fabricación.
can go ahead
Adelante,
and do the
pueden
study.
hacer ustedes el estudio.
But in order
Pero
to do
para
the
hacer el
study, of course, you
estudio
also
también
need
necesitas
funding. So we have
financiación,
already
así
submitted
que
a proposal to the FDA.
presentamos
The proposal
una
was
petición
reviewed
a la
reasonably
FDA,
well,
recibió una
but we
buena
have to submit some
respalda-
revisions.
bueno,
So we are
se ha
hoping
acogido
that
bastante
we're going to submit those
positivamente, pero
revisions in
tenemos que hacer
October
algunas
of this
revisiones,
year. And so
algunas
October of
modificaciones.
2025
Así que
is when we hope to get the
esperamos
funding.
conseguir financiación
So
en
we hope the
octubre
study
del
will start in 2026.
2025
Again,
para
I
que
wish
empiece el
we could do
estudio en
this faster,
2026.
but
Ojalá
yeah, the
fuera más
timelines for
rápido, pero
clinical
bueno,
studies is fairly long.
el marco temporal
But
de los
I do
estudios
want to
clínicos
describe the
es muy
clinical study to
largo.
you because I think we are at a
Estamos en
point
una
where we
fase en
can
que ya
make some
podemos
changes to the
empezar
study, and
a
that's what we
introducir
want some
cambios en
input
el
from
estudio,
you
porque aún no hemos
to see,
presentado los
and I'll
últimos
tell you the
papeles y
exact
por eso
questions on which
queremos
we want your input
la
to see what
contribución
do
de
you think,
todos
how
ustedes
should
para
we
que
do
nos
the
digan
study.
cómo creen ustedes que tendríamos que
So the
hacer el
idea
estudio.
is
La
that
idea
we'll
sería
start with initial
empezar
with the
con el
consent, so
consentimiento
the patients will
informado,
come
vienen
and
los
we'll test their
pacientes,
eyes to
se
see
hacen un
what their
examen
crystal
ocular
density
para ver
is.
cuál es
So,
la
so when you go to an
situación y
ophthalmologist, typically
normalmente
the ophthalmologist uses what
se
is called a
utiliza
slit
una
lamp to
lámpara
look in your
de
eye.
hendidura para
From
mirar
a slit lamp
el
picture, they can
ojo y así
calculate
se
how
puede
much
calcular
crystals
el número
there
de
is.
cristales.
And that's called the
Eso
crystal
la
density. The
densidad de
crystal density
cristales
score will be
se
obtained.
puntúa eso y
And then for
después
2
al cabo de
months,
2
we'll
meses
do
se
what is
hace
called run-in.
la fase
That
de
means you'll keep
introducción
using what you're
utilizando
already
el mismo
using,
fármaco que usan
just so we
para
get a
tener
good
una
baseline.
buena,
After 2
valor
months, we're going to
bastante.
put the
Después de
contact
2
lens in
meses
you.
se
We're going to have
pondrían
2 different
las
groups, a
lentes
low
de
dose and a high
contacto,
dose.
dos grupos,
And this study will
dosis
go on
más
for 10
altas
more
y dosis más
months.
bajas,
And
10
during the
meses más, y
study,
durante el
you will
estudio
be
se
essentially putting a new lens every
pondría una
day.
lentilla nueva cada día.
One question we want your input in, how long do you
Y
think
luego
you
una
should
de
wear
las
the
cuestiones es
lens?
saber cuánto
And
rato hay
there's a
que
survey
llevar
for
las
that.
lentillas. Hay
And
una encuesta,
we'll
les
give you this
daremos
link for the
luego
survey later, so that will be
el
one question that
enlace
is asked.
a la
Another
encuesta
question
para que nos
is that
digan
while
cuánto
this
tiempo
study is
creen
going
ustedes que
on,
había que ponerse las lentillas
would
y
you like to
cuánto
put
tiempo
lenses in both eyes?
creen ustedes que
Or
duraría
a lens
el
in one eye
estudio,
and
que
eye drops in
quieren
the other.
ponerse
The
una
benefit of putting
lentilla
lenses in
y
one and the eye drops in the
coliririo en
other
el otro,
is that you
por
have what is
ejemplo,
called
o
a
lentillas
control
en los
eye.
dos. Si se hace así
But
con
the
uno de
downside
cada,
for that
tienen
is that
una
it might be that the
cuestión control,
patients have to
pero
essentially still use 2
entonces
different products.
significa que los
And so it'll be good to get your input on
pacientes tendrán
what
que
do you
usar dos
think you prefer.
productos. Por eso pedimos su opinión
But
sobre
either
eso
way,
también.
then we'll have the study for 10
Estudio,
months.
10 meses,
After
al
1
cabo de
year,
un
the
año,
first
la
phase
primera
of the
fase
study
del
will be
estudio ya habrá
over,
terminado y
and then we'll
entonces
essentially
podremos
assess
evaluar
and see
y
which
ver
dose was more
cuál de los
successful. And depending
sistemas
upon which dose was more
funciona
successful,
mejor.
we're going to do what is
Entonces,
called an
y haríamos
extensional
una
phase.
fase
In the
de
extensional
extensión,
phase,
de
you just use a
ampliación, en
more
que se
efficacious dose.
modificaría la dosis,
And hopefully the
y
entire
ese
study will conclude in
estudio
about 3
acabaría en
years.
unos 18
And
años.
so 3
Y
years
3
after
años
starting,
después
we might go
de
to the FDA and try
empezar,
to get
podríamos
approval.
presentar la petición de aprobación a la
So
FDA.
that's essentially,
Básicamente,
this
esos
is
son
the
los
inclusion and
criterios
exclusion
de
criteria.
inclusión y
The only
exclusión. Lo
important
único
question here that
importante,
I
y
think we
creo
want
que
to
esto
we are still debating
aún
about
lo
is the
estamos
age
debatiendo,
of the
sería la edad de los
patients to start with.
pacientes a partir de la
We don't
cual se
want
puede
to
empezar. No
give
queremos
contact lenses to very young kids,
lentillas a
although
niños
ophthalmologists say that a kid as young as 4
muy,
years
muy
old can use a
jovencitos,
contact
aunque
lens.
algunos dicen que
And
a partir de
ultimately,
los
with
4 años ya se pueden
approval, we
usar.
think
Y no
we
sé,
will go
si
lower in
ustedes
age.
sugieren,
For the initial
pues
clinical
quizá
study,
más tarde para la
we're going to stay
aprobación
at
lo
about 8
hagamos
years
más
old. So
bajo.
we started with
Y
12,
empezamos
but now we have
a
decided to
los
actually
12,
go down to
pero
8. So this
inicialmente
study will be
pensábamos
available
12, pero
in the United
al
States
final
to kids who
bajaremos
are
a
8 years and
8,
older.
a 8 años.
So just to summarize, we have developed a product that can sustain the release of drug for long
hacer que
periods
el
of time.
fármaco se libere durante
We have
varias
shown
horas.
that
El
that product
producto
is
es
safe,
seguro,
it
lo
can
hemos
dissolve crystals.
demostrado, disuelve
We have
los
already
cristales,
proven
lo hemos
everything in
demostrado en
animals, and now
animales
the
y
human
ahora los
studies should
estudios con
hopefully start once we
humanos,
secure funding,
esperamos poder
because the FDA has already approved this
iniciarlos
study. So,
cuando tengamos la
with that, I would
financiación,
like to
porque
thank
todo
you
esto
all
ya
for your
está casi
attention.
casi
We'll link to more questions.
aprobado. Y con eso terminamos. Muchísimas gracias a todos por su atención.
Nuj, thanks very much for
gracias
an incredibly
por
clear and
esta
inspirational
charla tan
talk.
inspiradora.
Do we have any
¿Comentarios
comments from
o
the
preguntas del público?
audience? Yeah, down here.
Gracias
Thanks for the great work and also
por
the
esta
great
magnífica
presentation.
presentación.
My question
La
is,
pregunta,
what is the strategy for not using the preservatives with this pathway of using a lens so it won't
aquí
sting?
también habrá que usarse
not using the preservative, which causes the
el
eye irritation.
Great question. So this contact lens will not
no
have a
llevan
preservative.
conservante.
The reason you need preservative in your eye drops is when the eye drops are still in your bottle, it's sterile. It doesn't need a preservative inside because it was made sterile. The preservative is just to be there once you open the
pero una
bottle.
vez abrimos la
Now,
botella,
contact
pues
lenses,
no
once you
pasa.
open it,
Pero
will go
bueno,
into the eye, and the next lens will go into the eye.
la lentilla,
So because it's a
como es de un solo
single-day
uso,
lens, therefore there's
no
no
hace
need for
falta ponerle conservante.
Inside it's already
Dentro ya
sterile.
está
So there's no
estéril, no hay
preservative
conservante
because
porque
in the
dentro no se
inside you don't need
necesita.
preservative because it
Ya
was
se
made
ha
sterile.
fabricado en
And
condiciones
once
de
you open it like
esterilidad
this,
y
it goes straight
entonces
to the
como
eye.
después de abrirlo ya va directamente al ojo, pues no hace falta ponerle ningún conservante.
Angel, thank you
Muchas
so
gracias.
much. Is the downstream
La
idea
idea,
here, or let me just ask it like this: the
los
patients that
pacientes
already have a
que
prescription for
ya tienen que
eyeglasses,
llevar
right?
gafas, por ejemplo,
It's
está
contraindicated, for
contraindicado
them to have these eye
que
drops
les
and then throw contact lenses over.
que se
So
pongan,
we found in our
perdón,
population that
el
most, if
colirio
they
y
do
además se
have,
pongan luego lentillas. La
less than
mayoría
20/20 vision, and it's going to be
de
glasses.
nuestros pacientes que no ven bien tienen que llevar gafas
This
obligatoriamente.
contact
Entonces,
lenses,
con
could they
estas
actually be
gafas,
used for those
perdón,
that
estas
do not
lentes de
have
contacto
20/20
se
as well?
podrán usar para
Great
pacientes
question.
que
So
tengan
these contact
problemas de
lenses
visión.
ultimately
Estas lentes de
and
contacto.
Rachel, please feel free to jump in. Dr. Rachel
El
Bishop
doctor Rachel Bishop
helping us
nos
with the
está
clinical
ayudando con
studies,
los
so feel free to
estudios
jump in if
clínicos,
you
así
want
que
to
si
add
quiere
anything.
añadir usted algo,
But yeah, these
hágalo.
contact lenses ultimately will be
Estas
designed to
lentes
provide
de
vision
contacto
correction
también
as
se
well.
diseñarán para
Now, the reason
personas
contact lenses are
con
contraindicated with eye drops right
problemas
now
de visión, pero
is
ahora no
because
se pueden
you will
llevar
have
porque
to remove
tendríamos que
the
quitar
contact
la
lens, put
la
an eye
lente de
drop, wait for about
contactos,
5 minutes and
ponerlas
put it back
el
in,
colirio y
because you
después
can't put the
poner las
contact lens,
lentillas
you
otra
can
vez.
put an
No
eye drop on
podemos
top of the
poner
contact lens because the preservative in there actually can go into the contact lens
las
and
gotas
cause
encima de la
toxicity.
lentilla.
So yes, ultimate
Así que
approval,
sí, al
these lenses will
final,
be
cuando se
approved for vision
aprueben, se
correction.
aprobarán también para la
They could also
corrección
have photophobia
de
part in
la,
there. So they could actually
de la
block
vista.
some light
También
and they could also
podremos
address photophobia.
mejorar la
However, for the clinical
fotofobia.
study, we are
Pero para el
not going
estudio
to have
clínico
power
no
in the
vamos
lenses for
a
multiple
poner
reasons.
leches,
Rachel, do you want to
perdón,
maybe address that?
lentillas graduadas. Gracias por aclarar estas cuestiones. Una de las consideraciones es que la mayoría de los pacientes con cistinosis no llevan lentillas, como sabrán ustedes. No sabemos si las tolerarán bien o no. A veces, como la córnea ya tiene cristales, puede ser que ponerse una lentilla durante 2 horas sea cómodo, pero llevarlas 8 o 12 horas pues no sea tan cómodo. Yo tengo ojos secos, así que tampoco me va bien llevar lentillas. Para muchas personas, estas lentillas que son para el día entero, quizá no sea lo óptimo. Dicho esto, estas lentillas también te llevarán corrección si están cómodos llevando lentillas. Para muchos de mis pacientes son, no pacientes de cistinosis, sino los demás. Les digo siempre que se pongan lentillas siempre que estén cómodos, y si no, se puede poner un colirio hidratante o lubricante, porque son un buen tratamiento el lubricante. Y algunos dicen, pues yo prefiero llevarlas durante 6 horas y después me pongo las gafas, no sé. Uno puede ser usuario híbrido, no pasa nada con todo eso. Así que la pregunta es de si quiere usted llevar lentillas todo el día entero, sí, pues no sé, no lo sabemos, esto no lo hemos estudiado en pacientes con cistinosis, pero esta es una opción que espero que tengamos en el futuro. Hay varias razones por las que sería lógico que no se haga esto en las primeras pruebas con el primer grupo, porque por logística queremos separar los efectos del tratamiento, los resultados del hecho de que se lleven las lentillas durante el día entero y eso sea incómodo, quizá porque no les vaya bien llevar lentillas tantas horas, o quizá porque el fármaco les molesta. Esto es una respuesta que les parezca lógica, por eso no queremos perjudicar la limpieza del estudio. Además, estamos en el Reino Unido, esperemos que este estudio también se pueda llevar a cabo en Estados Unidos, y soy sensible al hecho de que no se puede uno subir al avión y participar en el estudio, pero como saben ustedes, por la historia de los fármacos, Si esto funciona, esperamos que sí, esto luego también es un estudio en el que les pediremos que participen ustedes más adelante, y también ahora que ustedes respondan a la encuesta que hemos puesto y que ofreceremos más tarde, pues si están ustedes motivados para hacer su aportación, fantástico, y si no lo desean, pues no lo hagan. Simplemente poder compartir la investigación nos parece a nosotros ya gratificante, y tener feedback de ustedes, pues muy bien. Y hablaré más de la cistinosis y de los ojos mañana cuando hablemos de cómo mejorar los tratamientos actuales.
Thank you very much. I think we've got another question down to the left here.
Hi, some of my questions were answered there. One of them was to do with when or how can we get involved, but I'm guessing as a UK citizen,
aunque como vivo en el Reino Unido, creo que no podré porque se va a ser llevado a cabo en América.
Could this, I was aware, as my understanding, I've been told before wearing contacts could be quite difficult because of the crystals in the eyes. That was my understanding. Is that like, how would you get around that? And could it be, if you're saying could be worn for a few hours, could potentially be used in conjunction with a drop? Perhaps you wear for a few hours in the day and then a drop in the evening if you can tolerate for long periods.
So excellent question. My hope is with the amount of drug being delivered by this contact lens as I anticipate, I do not think you would have to use cysteamine drops in those other hours of the day. You might need lubricant drops if your eyes feel a little dry, but the hope is this is a complete replacement for cysteamine eye drops.
Pero en realidad no es el objetivo.
I'm presuming then the lubricant wouldn't affect the cysteamine.
That's correct. So in general, what we say with eye drops of any sort is when you put a medication eye drop in the eye, you wait 5 minutes before you put a lubricant drop. Let the eye have a chance to absorb. And that philosophy gives you a very good demonstration of how much time we actually think that drug is having contact with the cornea. I will add, and this is just in the hypothesis zone, it's quite likely that wearing this contact lens even for an hour of continuous drug exposure to cornea is way more interaction with the cysteamine crystals than putting drops in even 16 times a day. If it's 5-minute contact time, even 10 times a day, 50 minutes, we don't know these answers, but it's quite possible that this constant dosing actually means maybe you wear it an hour a day eventually when we understand it better. You may recall the cysteamine eye drops that came out of Cysteran in the United States in 2012 were studied hourly while awake. Right, so the FDA approval label says use hourly while awake, 14 times, 16 times a day. Of course, no human can really do that. So we have to pick something, and we start from there. And perhaps we find out, hey, you wear this for 6 hours every 3 days, or I just don't know. But our starting point is the best guesses we can make at this time.
Thank you very much. I'm going to have to call this session.
Actually, if I can just show the last slide about acknowledgement. Sorry, I forgot that. I really want to acknowledge Cystinosis Ireland for providing a lot of the funding for this work. In the last cycle, they also joined the Cystinosis Foundation UK. So,
quería también señalar el Cystinosis Ireland y Cystinosis Foundation del Reino Unido como reconocimientos.
Thank you very much, Anush. Incredibly exciting. Now, the next talk, if I'm correct, looking at the IT people here, is actually a video. Is that right, from Dr. Paul Godier?
El video del profesor Paul Godier.
Am I saying a yes?
Sí, ¿verdad?
And so that should appear on the screen. Paul was due to join us here. Many of you will know Professor Godier. He's Professor of Pediatrics at McGill in Montreal. But you'll also have heard of the cyberstrike issue last week, which influenced all of the routing of the aeroplanes, and he was caught by that. So it does demonstrate how much of an integrated world that we live in. And Paul's very kindly agreed to produce his talk, and is going to show it.
Now, the motivation to embark on this is, I think, captured nicely by this slide from Dr. Baudin Sartorius 12 years ago. This was a study in which a group of people with cystinosis were, their natural history was studied. And on this slide, what you can see is that on the y-axis, the left vertical axis, is plotted the age at which an individual with cystinosis develops end-stage renal disease, ESRD, or time for dialysis or transplantation. On the x-axis beneath is the age at which an individual started cysteamine therapy, oral cysteamine. So if we focus first on the right-hand side of the slide, the lower quadrant, this is a group of people who never received any cysteamine. And if you let your eye drift to the left over to the y-axis, you can see that they went into renal failure at about the age of 10. Not very good. Now, if you look at the left upper quadrant, marked in blue here, you can see that there are a sizable number of people who started cysteamine therapy before the age of 3 in this study. Those people really had a big benefit. They went into renal failure not when they were 10, but in their 20s. And these people at the top of the bar here are open circles. They have not yet gone into renal failure. They're up to 28, 29 years of age. So fantastic. This is a therapy which has made a big difference for everyone with cystinosis, and it was discovered 40 years ago. Now, the disappointing part to this therapy comes from the fact that even though these people clearly have had a benefit, nevertheless, they are projected to go into renal failure at some point. So it's not a cure, it's a delay, partial improvement. And the other observation we can make from these data are the fact that in this red circle on the left lower part of the graph, you can see that about a third of the group who, although they started cysteamine therapy before the age of 3, they have not benefited very much. And the average age at which they're going into renal failure is very similar to those people who never saw any cysteamine. So we need more. There's something we're missing. One possibility is simply that the people who don't benefit from cysteamine therapy are unable to tolerate it, and so they don't get enough of the medicine. That's possible. But scientists have suggested, a lot of research has indicated that there may be some other explanations. So I'd like to go after those explanations briefly with this little cartoon. So this is a picture of a real proximal tubular cell, I think from a mouse. And you can see those little finger-like projections from the cell projecting upward into what is the urine flowing by. And one of their jobs is to capture proteins that have drifted into the urine and not let them escape. They're all good nutrients. And so those little finger-like projections will capture them, encase them in a small vesicle, and send them down into the lysosome. They're chopped up into the amino acids. The amino acids get out of the lysosome and get back into the cell to be used to build new proteins. And there's another system depicted on this cartoon, labeled AP, and that is also a set of small vesicles that actually go around the cell, cleaning up debris, unused mitochondria, like these dark structures in the cell, damaged mitochondria, or proteins that are no longer functional. They gobble them up, and they again come to the lysosome, dump their cargo, and then leave. And again, those proteins are chopped up. So there's a huge amount of cysteine among other amino acids that is produced inside the lysosome. And the cysteine, like all the other amino acids, has to get out to be used. And the trouble in cystinosis is simply that the gene encoding that little channel is faulty. Now, cysteine builds up, destroys the lysosome, destroys the cell, so it's not good. And what happens with cysteamine is that cysteamine gets inside the cell, inside the lysosome, chops the cysteine through a chemical reaction into parts, and each of those smaller little subunits can get out through another alternative channel, the back door, so to speak, even though the primary channel is messed up. So that's the way the cell works. The other fascinating thing, unfortunately, is that there seem to be functions for the protein encoded by CTNS, which are not just the channel that allows cysteine out of the lysosome. These other functions seem to be necessary for the capture of proteins that I've told you about and the capture of debris from inside the cell. And those processes, if cysteine is not there, are faulty, and there's nothing we can do about it by giving cysteamine. So that sort of thought process has always made me think that in the end we're going to need something to put back the normal cystinosis protein that is needed for many jobs. And when I need a bit of inspiration, I simply look at this picture of a bunch of kids, amazingly delightful kids, at a cystinosis meeting down in the United States, and I envision that they're all smiling, but they're probably saying, "We're hopeful, we're hoping for a new leap forward. It's been 40 years, after all, since cysteamine was discovered. We need something more, and we need to find it." So, my low point, perhaps, in this long time journey of my own was during the COVID pandemic. And my research program had to stop, and one of our projects was completely halted. And of course, for all of us, research or no research, for all of us, the pandemic brought sort of a bleak future. Sort of like this picture of the Western Irish coast on a bad day. Nothing really brilliant to look at. But across from where you're sitting, in Manchester, at the Manchester Art Gallery, is a picture of this fellow, John Milton. He was a famous English writer, and he is said to have penned, among many other things, the phrase, "Every cloud has a silver lining." And of course, he was referring metaphorically to the sunlight wrapping around the edges of this dark cloud, indicating that there was something better coming along. Now, for me, the silver lining to the pandemic came in the form of this fellow, Paolo Martini, who was the chief scientific officer for rare diseases at Moderna. He came to McGill to speak to us, and of course he talked about the mRNA technology that was used for the COVID vaccine, but he also mentioned his idea that it could be adapted to treat rare diseases. I had the fortune to have dinner with him, and of course what I said was, well, so could we use this for cystinosis? And he said, why not? There's some work to be done, but it's possible. Now, what was the crux of the idea? As you know, I'm going to do a little diversion for a moment to explain how the COVID vaccine was produced. And as you know, our DNA contains genes. Each gene has the code that can be copied onto an RNA, which then produces the protein. Now, the Moderna scientists and the Pfizer scientists knew the code for these little spikes that stick up from the COVID virus, the spike proteins. And so they plugged them into their computers, typed out the mRNA code, and then took that, they synthesized the RNA artificially, stuck it into a lipid droplet called a lipid nanoparticle, LNP. And they took that mix of things, shook it up in a vial, stuck a needle into the vial, pulled a little bit out, and shot it into our arms. And that was the COVID vaccine. Now, what happened in the minutes and hours and day or so to follow was that stuff floated around in our bodies, and when it came in contact with certain cells, the cells would gobble up the LNP, bring it inside the cell, cause it to open up and discharge its RNA, and that RNA then was available to bind to the work table for these things, the ribosome, and produce the protein that was encoded on that mRNA. And of course, in this case, it was that spike protein. Now, when the immune system saw that, the immune system would say, "Whoa, this is foreign protein. This is not right," and form an antibody against it. And that was the basis for the protection. How would this work for cystinosis? Well, in somebody with cystinosis, nephropathic cystinosis, there would be two copies of the CTNS gene, and both would be faulty. So they were unable to make the proper RNA to make the protein that would keep the cell healthy. So I proposed to Moderna that they would punch out the proper code for CTNS mRNA, stabilize it in their special way that does not change the protein at all, but makes the RNA last longer, package it inside an LNP, shake it up in a vial, stick it into people's arms, and hope for expression of CTNS. The idea is that the particles would be taken up by our cells, released, the RNA released, and when this RNA gets onto the ribosome, it doesn't make COVID, of course, it makes CTNS, the cystinosin protein that we need in our cells. Now, can this strategy really be applied to humans? A major leap forward really came from the Moderna rare disease group themselves. This is a paper reported in Nature about 4 or 5 months ago, and it was an interim analysis of their first human clinical trial. They were studying the mRNA therapy that was being given to people with a rare disease called propionic acidemia, not cystinosis. But this clinical trial began back in 2021. They actually used 2 different RNAs to produce 2 subunits of this enzyme that was needed. They recruited 16 people, kids. They followed them for up to 1 year, and they gave them different amounts of the RNA. And they had 15 centers spread across Canada, the United Kingdom, and the United States. So this appears to be promising. We haven't seen the final results, but it's going along and seems to be going smoothly. So there's hope, I believe, on this basis. So I decided to go back to Moderna, discuss things with them, and Paolo Martini put me in touch with this lady, Megan Brundage, who's a scientist at Moderna, and so she helped me get this ball rolling. With Moderna's support, I then applied to the Cystinosis Research Foundation in California, and was awarded a grant to pursue this. And funding began in January, so this is relatively recent. The first job I had to do was to figure out the sequence that I would ask Moderna to produce. And so shown here in the blue circle, the classic cystinosin protein, the RNA code, is very well known. And this is the one that winds up in the lysosome, in the membrane of the lysosome, and acts as a channel to get cysteine out. No question we had to supply that sequence. But to make things a little bit more complicated, thank goodness, long ago, Anna Taranta and Francesco Emma from Rome discovered that there was at least one other form of cystinosin protein, or CTNS mRNA, and it had a longer tail on it that stuck into the middle of the lysosome than the usual one. Everything else is the same, but it had this long tail on it. And it looked as though this accounted for 15% of the total cystinosin protein in the cell, and it seemed to me that it was foolish to think this was doing nothing. And so I decided at that time that we really would have to consider both mRNAs as part of therapy until we knew more. And in fact, Anna and Francesco had spotted the fact that this other form of cystinosin appears a little bit in the lysosome, but also very prominently at the surface of the cell and associated with some of the little vesicles spinning around in the cytoplasm, some of which may or may not be the particles that are transporting proteins from the surface or transporting debris into the lysosome, as I mentioned before. So I just felt that we could not ignore this and take a guess that the main form was the only thing that would be needed. So I went back to Megan Brundage, explained to her the whole thing, and said, we need you to make an mRNA for both forms because we need to test them both. Furthermore, we need to follow them when they get into cells, and we need to be able to watch over them. So can you stick on some tags? And she said, no problem. I've got plenty of little tags I can type into the sequence and put them at the beginning or the end or wherever you want. And you will be able to track the product, protein product that comes from the RNA that we're going to give. I said, we'll take them all. We'll test them all. So she sent us the first RNAs in March, and we began to test them in April. So what we did is outlined here. We took those RNAs that Megan had sent us with little tags on the end, two different types of cystinosin, and a little tag on one end or the other, and we artificially just forced them into the cells in the laboratory, and we used human kidney cells from the proximal tubule. These cells are very well known to everyone, and we use them to do the job. So we then began to encode. Well, we smashed up the cells. We took the proteins that were inside those cells and spread them out on a gelatin slab. And then we exposed that slab to an antibody against the tag, V5, so we could find our protein wherever it was. We could even figure out how big it was. And sure enough, we found what we wanted. We found a main band right at the right size, and it reacted with an antibody to this tag, so we knew that was the real stuff, the real McCoy. Then we did the same experiment and said, well, we know it's the right size and it's the right protein, but is it going to the right place? So this time we analyzed the cells with a microscope. In this slide, we're looking down through the microscope to see the red color that represents our little tag on the molecule. So when the molecule is produced, we'll be able to see this red tag. And there they are. They're little blobs of red inside a single cell. But is it really in lysosomes? And the fact is that they are, because we stain the same cells with another colored antibody that would detect something on the surface only of the lysosome. And so if you look carefully, every blob of red corresponds to a blob of green. And you only see the red stuff where you see the green stuff. So this does exactly what we wanted it to do. It goes right to the right spot and nowhere else. And just as predicted by Anna and Francesco, the other isoform, the other form of cystinosin with this long LKG tail, appears at the surface, that line running along the outside of a cell, as well as in some of the little vesicles inside. So this is exactly what was spotted before. And so we think that both forms of the cystinosin that was produced by mRNA, the CTNS mRNA, both forms are correct. Sorry about that. So one final problem. We think we have the tools, the mRNA tools, to do the job, but there's one more task that is an important hurdle, and that is that the LNPs, the delivery system for these mRNAs produced by Moderna, they love to go to the liver. The cells of the liver just gobble them up. Nearly 100% of the LNPs get into the liver. And the problem is that the liver is not much of a problem in cystinosis. We really need to see the RNAs delivered to the kidney, the muscle, and the endocrine organs. So that was a problem. And I began talking to Moderna about this a year ago. And they, just 1 or 2 months ago, sent me this information. What they did was to take their standard LNP, lipid nanoparticle, and they made RNA for this fluorescent protein, GFP. And so they packaged the RNA in there, and then they injected it into a mouse. They took out the kidneys, and they did this analysis. And what we're looking for, forget this big red blob and that green blob, we're looking for another green blob that appears at a smaller size, which is expression of GFP. And oops, the standard LNP that worked beautifully for making a COVID vaccine doesn't deliver to the kidney. They had other formulations that they tested. So LNP2, nope, there's nothing. They tried all sorts of tricks and nothing in the kidney. They tried another one. They had some new ideas, but no, nothing at all. And then they came across a most recent novel, a new formulation of the lipids and things in the coating of this lipid nanoparticle, the amount of cholesterol and so forth. And it was a gold mine. They found that here's that signal that we're looking for, that's GFP, and it was seen only when the mice were injected with this formulation, which I call now, we agreed to call Charlie, because that was the name of the dog for one of the Moderna scientists, Mike Zimmer. And so we agreed just to refer to it as Charlie, because Moderna does not want to tell me exactly what's in there and what the name of it is, their name. So, but in speaking to Mike Zimmer, we agreed that this looked really good. We're going to package in the future the CTNS mRNA into this thing, try to get it to kidney, and then we're in business.
al riñón
So,
y lo teníamos hecho.
with that, I'll quit. I think that that this project looks very interesting, and it looks like this fjord down in the bottom of the South Island of New Zealand. It's so interesting. It appears to hold such promise that we just have to go in, take a look at this, and see what's there. It was actually a fjord that Darwin sailed into. He discovered some flightless birds and some other stuff around the corner of this very fjord, and it helped him to develop his whole theory of evolution. And so I hope that our foray into this fjord, or metaphorically, will bear fruit. These are the people who are actually doing the work in my lab on this project. These are my collaborators. And my wife, Elena Torban, is actually a co-PI, principal investigator on this project. Mike Zimmer and Megan Brundage are from Moderna. I talk to them about every 2 weeks. And of course, this would not be possible had it not been funded by the Cystinosis Research Foundation. So I thank them for taking a chance on this project and funding it. I hope that it will deliver in spades. Okay. Thank you very much.
Muy bien. Pues allí tenemos más elementos para el futuro. Es fascinante ver cómo los avances en un ámbito de la medicina y de la ciencia pueden tener tanta repercusión de cara al futuro. Esto nos lleva al último orador de esta sesión y vamos a pasar del laboratorio y de los elementos de la ciencia a otras cosas que son igual de importantes, pero más directo, ¿no? Cómo se organiza la prestación de servicios para usuarios, los servicios que nos parecen a nosotros óptimos para los pacientes con cistinosis. Y voy a presentar al doctor David Game, que es un amigo y compañero. Aquí lo tenemos. Y él es nefrólogo consultor en el Hospital de Guy's en St. Thomas y ha sido una persona decisiva en la creación de centros específicos de referencia de cistinosis en el Reino Unido. Tienes la palabra, David.
Thanks very much, Graham. So, some fantastic talks just then. I don't know if everybody else's scientific brain is a bit saturated with science, but mine definitely is. And I think if you followed all of that and you're still going, then you could probably answer at least a degree level question on molecular biology. So it's really great talks and very illustrative and some really inspirational work for the future. So don't worry, I'm not going to talk about mRNA or anything like that, even if it's your thing. I'm going to talk
mRNA ni nada por el estilo, sino de cómo se ha organizado la atención sanitaria desde Irlanda y desde una parte bastante adulta de la cuestión, porque soy nefrólogo de adultos, pero eso también obviamente implica a los nefrólogos pediatras.
So I'm going to talk about these national designation centres, but I'll talk about the word hub or hub centre or multidisciplinary clinic. I'm meaning those things are all interchangeably going to be the same thing. We're going to talk about how these hubs were set up in the first place, how they look now, a little bit about transition between paediatric and adult services. We'll tell you a bit about radar if you don't know about that already, but I'll tell you about that. And then some thoughts about how we might take these hubs forward. And I'm not going to take the whole half hour, so we'll definitely
está llevando adelante, así que bueno, no tardaré media hora, o sea que pronto podrán irse a tomar el café. Gracias a la Fundación para la Cistinosis del Reino Unido, que están implicados en esto desde hace mucho tiempo, 10 años, y como New Jonathan, como sabrán,
were instrumental in setting
murió
up the foundation.
hace un tiempo, pero
As you can see,
él
he had
y
cystinosis and
su
had an
padre
MBE for his services to charity.
fueron personas decisivas para crear esta organización y recibieron
nobody in this room needs persuasion
unas
that people with
condecoraciones por
cystinosis
ello.
would
Todos
probably
sabrán,
benefit from
seguro,
going to a one-stop
y estarán de
clinic
acuerdo
rather than going to a million
en que
different
las personas
clinics,
con
one after the other,
cistinosis
one after the other,
prefieren ir
different days
a
of the
una
week,
sola
different
clínica
hospitals,
que a no sé cuántas
different
clínicas
specialists,
distintas
some
en
of whom
distintos
don't know that much
hospitales
about cystinosis. And so,
para hacerse pruebas,
we've
muchas
decided
veces
we
con
try and put
profesionales que no
some
saben mucho de
thoughts together.
cistinosis, así que decidimos
Those of you that
organizarnos
commented
juntos
on
y
how long everything
bueno,
takes will see what's happened here. And
todo tarda mucho.
it's
Hace
10
10
years ago
años
that we had a meeting of a few
nos
people
reunimos unas
in
pocas personas en
England, I
Inglaterra,
think it
creo
was
que
in
fue
Birmingham,
en Birmingham,
and
y
we kind of had this core writing group
empezamos
and
con un
wrote a few
grupo.
things together, and you can see all the different
Vemos aquí los
hoops, and
distintos grupos.
Elena talked about all
Elena ha hablado de
the
todos
problems in
los
trying to get drugs
problemas
through to market
para
these days,
intentar que los fármacos lleguen
it's
al
not
mercado,
dissimilar
y es algo
to clinics
parecido a
or
lo que
anything
pasa en la
that you want the
clínica.
NHS to pay for.
Y
It's
bueno,
really very difficult,
todo
and
esto es muy
they put
difícil,
lots
se
of
plantean
barriers in the
muchas
way,
barreras,
probably
seguramente de
intentionally, I
manera
don't know, and you
intencionada,
have to just
y
go through
uno
each
tiene
one
que
methodically and
ver
try and persuade
esas
people that
cosas
you're doing the right
metódicamente para
thing.
intentar convencer a
And
todo
so
el
you can
mundo
see from here, from the core
de
writer group in
que
June 2014,
tienen
then we
que
had
hacer
the
bien las cosas.
consultation,
En
service
junio de
specification, which I'll tell you
2014,
about,
pues ya se
and
empezó a
then
formar este grupo,
then you get
este
the
equipo de
tendering process, and
redacción,
then you put
primero
your
con las
tender in, and
especificaciones,
so actually it
después
wasn't quite 10 years
se
because it
hace
was
pues
autumn
una
22 that the first clinics
definición
were
de
officially
requisitos y
started,
necesidades, luego la
and we've
licitación,
hopefully
etcétera,
made some
etcétera, y bueno, pues
progress
hemos ido
since then.
avanzando en todo
So
esto.
sorry guys, it just takes a long time to do stuff in healthcare. And this is the model that we based the whole thing on. This was drawn up fairly early together with NHSE. And what we're trying to show is that it's a hub and spoke
aplicar
model,
un
right?
modelo
The hub is where
centralizado
the adult and pediatric
con un centro
centers are.
y
It's a
con
multidisciplinary clinic,
pues
so
varias
hopefully
cosas
just
más,
one stop for
esperando que
everybody.
sea con una serie de
Everybody, hopefully, that you
ramificaciones,
need
¿no?
to
Para
see
que
in one stop,
todos
but
puedan
actually
verlo
the
todo,
delivery of
o
quite a
todas
lot
las
of this,
pruebas y
maybe
todas
all of
las
it,
visitas que
depending on
tengan
where
que
you
hacer
live,
se
will be
puedan
local.
hacer en un
And the
único
idea being that you
lugar,
identify the need
y con
here,
alguna esperanza de que pueda ser
cajole the local
local.
people to
Se
do
tiene
what
que
they need to do,
intentar pues que
and
todo el
then try
mundo
and follow
pueda
up
hacer lo
to
que
make sure
necesita
that
hacer
that
en
is
un solo
actually
sitio y que
happening.
todas estas consultas se puedan realizar
Obviously, you
conjuntamente.
have
También
other
hay otros
specialists involved, quite
especialistas que
apart from the
están
MDT.
más lejos,
Clinical
pues
genetics will be involved in
hay
parts of the journey.
genetistas en algunos
Obviously, we need to measure the white
aspectos,
cell
porque habrá que
cysteine,
pues
and then there's preconception
también
counselling
hay
for some people, and other services that
asesoramiento
will be brought into the
prenatal
whole model. And
todos
everybody should
deberían
be having at least an annual
tener
review in the
una
hub. That's what
revisión
we
anual
said
como
we would
mínimo
do.
en este
Some people
centro de
have more needs
referencia.
than
Algunas
others and see
personas
much more often, and
necesitarán
others have
pues
very
más
few, and
visitas que
we'll be
otras,
seeing every
algunos solo
year.
tienen que verse una vez al año, etcétera, etcétera.
And so the
El
specialists that need to
especialista
or
que
should be
tiene
at
que
the
existir
different
en las
clinics
distintas
will be different
clínicas, en
between
las
adults and
diferencias,
children,
sabemos que hay
adults and children,
diferencia
obviously.
entre niños
And so this was the list that is drawn up in
y
the service
adultos. Esta es la lista de
specification. You can
especialistas
see the
que
whole thing
vemos
online if you want
que
to.
se
It's kind
establecieron
of a
en
contract between
el pliego de
NHS
condiciones. Este es
and the
un
hospital that's
contrato,
delivering
está
the care.
está en línea,
And so
figura en
you
el
can see
contrato
that
entre el
the,
hospital o
so
el
in Peds,
centro de
for example,
referencia y
you'll
el
have
Servicio
some,
Nacional de Salud. En St. Pitt's, por ejemplo,
some genetics, for
tenemos
example, that may
genetistas
assist
pues,
with
que pueden ayudar
diagnostics unless
con el
you're also
diagnóstico,
using white cell
también que
cysteine.
pueden estudiar la cisteína
Pregnancy
en
in
sangre,
pediatrics probably less required.
pediatría menos
Pharmacy for everybody.
necesario en todos los casos.
Physiotherapy definitely important
farmacia
in
siempre es
adults because
necesaria, ayuda nutricional también para todos.
syndrome, if you like, is
El
more
síndrome
common
muscular
as you
es
get
más
older.
necesario cuando
And so
uno es
different things
adulto.
required at
Así que hay
different times. And
distintas
so that's what
necesidades en
we're
distintas
supposed to be
épocas. Por ejemplo,
delivering.
para eso se necesitaría mucho más fisioterapia, etcétera, etcétera.
And so the tenders went out. The hospitals
Los
put their
hospitales
bids in,
hicieron sus propuestas
and the way
cuando
it was
salió
formulated eventually
la
by NHS England was
licitación
that
y al
you had to
final
put a
pues se
bid in
tenía
with
que
jointly between a
crear
pediatric centre and an
una
adult centre. So when
licitación
they
en la
first put it out, it
que
was going to be a bit of a free-for-all.
se
but then they said,
combinara el
no, we want
servicio
you
de
to go in
pediatría
together, so
y
we
servicio
can
para
ensure that
adultos
the
para que el
transition
servicio de
process
transición
is good.
funcionara bien. No se
And so,
podía ir solamente
naturally, we
pediatría
went
o
in with
solamente
the Evelina
adultos.
in London
Y
because that's based at my
Evelina,
hospital.
mi
And so
hospital
and
y el de
then
Bellina fueron los que se combinaron
and
para
Birmingham
Londres.
Children's, and then
En
Manchester
Birmingham, pues el
Children's
universitario
also
y el de
awarded the contract.
pediatría.
So I've given some
Aquí ven
pictures,
algunas
the nicest
imágenes, son
pictures I
las
could
imágenes
find
más bonitas
of
que
the
podía encontrar de estos
hospitals.
hospitales, pero bueno. Y
Guy's and St Thomas, it's two hospitals, but this
este
is
es el
St
de
Thomas's
St.
hospital
Thomas.
here, and that's where
Guy en
the
St. Thomas son dos,
centre is,
y
where
este
we
es
have our both adult
el
and
lugar donde
pediatric
tenemos la
clinics.
clínica de adultos y de pediatría. en rojo. La idea es que todos los pacientes de Inglaterra hagan seguimiento o tengan seguimiento en una de estas tres clínicas. En muchos casos no será, sobre todo, quien otorgue o quien realice el tratamiento, pero sí que serán los que supervisen el total. Eso es un poco la idea. Ese es el plan que tenemos, aunque luego una parte de las actividades se hagan en otros centros. También se pueden ver pacientes de fuera de Inglaterra en estos centros de referencia. Eso se puede hacer. En Escocia tienen un gran nefrólogo fantástico, en Glasgow que también tiene una clínica para ello. El propósito es ayudar a los pacientes con cistenosis y a sus padres y a sus familias y cuidantes para que todos reciban una atención de calidad. Lo que pasaba antes es que unas personas recibían un servicio de muy buena calidad, otros no, y queríamos que todo fuera lo más equitativo posible. Todos tendrán revisiones anuales. Si hay problemas con el transporte, también hay mecanismos dentro del NHS para que se reembolsen los gastos de transporte a estas clínicas. Aquí vemos imágenes del hospital de St. Thomas, el centro de enfermedades raras, también hay otros, y la idea de este tipo de centro es que esto sea algo más bonito que en los centros de trasplantes o los centros de nefrología, que sea más agradable a la vista, más acogedor. A veces, con suerte, te dan un café o un té y que haya pues, más información sobre las patologías, en este caso sobre la cistinosis. Y en general, pues, muchas veces hay varias salas y en función de donde esté la enfermedad rara, pues los pacientes van entrando y saliendo. Hay dos montajes, o el paciente en una sala y los distintos especialistas entran y salen, o al revés, los especialistas en una cada uno en su sala y van entrando y saliendo los pacientes. Bueno, siempre hay que buscar la manera óptima porque depende un poco. Y aquí vemos algunos pacientes. Esta es una imagen de la clínica oftalmológica. Está muy cerca, debajo, del centro de enfermedades raras. En nuestro caso, pues van a la planta de abajo, se hacen las pruebas y suben. Para fisioterapia, a veces hay algunos dispositivos increíbles para las personas que tienen debilidad muscular. Es fabuloso lo que se inventan en nuestras clínicas. Esto se llama el SNP test. Esto sirve para medir la respiración. No sé quién ha hecho un SNP test. Es como una especie de espirometría. Uno respira y tiene que respirar la fuerza de la musculatura respiratoria porque te tienes que tapar el otro agujero de la nariz. Mi compañero siempre dice, vale, muy bien, venga, va, venga, pues hacen toda esta juerga diciendo, hay que intentar inspirar y expirar con toda la fuerza posible para demostrar la fuerza. Es importante que hay que verificar estas cosas porque nosotros siempre queremos pensar que detectamos las cosas, pero si las detectamos antes podemos intentar pues o ralentizar el proceso o mejorar las posibilidades de tratamiento. Eso es una resonancia. Tenemos un umbral bajo y podemos hacerlas con bastante facilidad a las personas que tienen cistinosis.
So the hubs provide specialist medication, so you've heard a lot about that today. And so the hubs will be, we can arrange to have Cystagone and Cystadrops delivered to your house. We can arrange for prescription of other medication, including the Fanconi stuff and also for the patients that don't do well on Cystadrops, we can try and help you with the Cystadrops or prescribe the preservative-free stuff from the aseptic pharmacy.
Pues fármacos sin conservantes, colirios sin conservantes.
Radar is a rare disease register, which I've got a slide on in a moment, and we submit data to Radar, more data than is required for the basic registration of people with a rare disease, because we're using this as kind of our
en el radar, que es el registro de personas con enfermedades raras, y usamos toda esta información para hacer seguimientos.
And I said the hubs are integral to further applications to NHS England such as proposals for other treatments such as ProCIS.
Y bueno, estos centros también están abiertos a que se propongan
So in terms of transition, so
más
moving from
tratamientos para
peds
otras
to
enfermedades. En
adults,
cuanto a la transición entre la atención pediátrica y adulta, los niños
may well
hacen la
transition
transición a su
partner heart
unidad
unit, for example.
para
So,
adultos. Nuestra clínica de pediatría está se hace por la mañana, la de la tarde es para adultos. Si tienes que hacer la transición, pues te quedas a comer. Pero si uno viene de la universidad o donde sea, pues no hace falta estar allí, puede ir a cualquier parte. Si tiene que irse de Londres e ir a Manchester, pues se puede trasladar a Manchester y allí en esa unidad le atenderán. No tenemos ninguna preferencia al respecto.
Adults who are not
not
currently under a pediatric centre can then basically go wherever they want. We don't mind. We won't take it personally. If you live in London but still want to go to Birmingham, that's up to you. We really don't mind, as long as somebody
Lo que importa es que haya alguien que supervise todo el tratamiento. Y luego,
recruitment is a bit of an issue. So if you're not currently under a hub centre and you want to be, then we can easily sort that
inscrito en un centro de referencia,
written to all the chief executives of hospitals, and
podemos escribir pues, las cartas que corresponda a
it sits in their desk and then it gets emailed to various people and cascaded and nobody reads it. And so that's not the best way to try and recruit patients, I would say. And referrals are by conventional
muchas
email
veces nuestro reclutamiento lo hacemos a través de las redes de personas con enfermedades renales, compañeros, entidades de beneficencia, etcétera, etcétera, y también con referencias a través de este correo electrónico que veíamos en pantalla. La transición de un paciente con cistinosis no es fácil, porque aunque la atención renal la den en hospitales pediátricos, la atención de adultos se puede ofrecer en todas partes. Así que si uno hace la transición desde el Hospital Pediátrico de Londres y le hacen la diálisis en el centro de Londres, pero uno vive en Brighton, la diálisis se la harán en Brighton. Así que hay toda una serie de arreglos para poder hacer estas transiciones y que la atención funcione mejor. Tenemos que garantizarlo formalizando mejor los procesos de transición en las distintas especialidades que exigirían una transición. Así que si uno tiene distintas necesidades, habrá que hacer la transición a los servicios locales, pero siempre bajo la atenta mirada del centro de referencia para que todo se haga bien y se haga todo. A veces hemos tenido algunas dificultades, pero hemos tenido algunos problemas y sabemos ya más o menos lo que tenemos que hacer. Luego, ¿qué hacemos? ¿Cómo sabemos que estamos haciendo lo correcto? Pues porque nos comparamos con el público internacional, como aquí tenemos un público internacional, comparamos lo que estamos haciendo en nuestros centros de referencia y lo cotejamos con las guías clínicas más exigentes. Esta es una revisión que se publicó hace varios años y aquí habla de todas las revisiones que tienen que hacerse las personas con cistinosis y aquí no hace falta que veamos pues las distintas tablas, etcétera, pero aquí este es básicamente un recordatorio de todo lo que tendríamos que hacer y nosotros pues lo hemos revisado y casi que sí, que casi todo. No tenemos atención dentaria porque es imposible en el servicio de salud británico ni dermatología, pero la mayoría de los pacientes sí, a los que se han hecho trasplantes de riñón sí lo tendrían y se les puede derivar si fuera necesario.
This is a website that anybody in the UK probably should have a look at because it's got some good information about the hubs and other things regarding cystinosis. It's hosted by the UK Kidney Association, and it's got patient information, clinician information, and then a dedicated page for information about the hubs.
Sobre los centros de referencia.
And what you will see on that page is some contact details for the different hubs
de contacto de las distintas centros de referencia de Inglaterra y si no les han derivado a un centro de referencia, pues tendría que hacer en principio su médico de cabecera, pero si no lo consiguen, envíennos un correo electrónico, pongan esto en Google y estaremos encantados de ayudarles. Estamos aquí a su disposición. Así que ahora en estos momentos,
these are the numbers of patients under follow-up. You can see in the different units. There are cohorts of patients who are still under other hospitals that we all know about. We're just pulling these patients into the hubs at the moment.
Para que se incorporen al centro de referencia, pero esperemos que dentro de poco, la próxima vez que nos veamos, pues que las cifras hayan crecido y que todo el mundo esté siendo controlado, o al menos bajo el radar del sistema, ¿no? Pero por las recetas de dentista, sabemos que hay muchas personas que están
So this is RADAR. This is the UK Rare Renal
Este es el
Registry, and it's
sistema
all
de
millions of different
enfermedades
rare
renales raras
less than
del
100, but
Reino
quite a lot of rare
Unido.
diseases,
Es
all
un
that are
registro
registered
nacional, hay
for
muchas
data
enfermedades
collection, really, and
raras,
it's
es
anonymous.
anónimo, pero
There's no
sirve
risk
para
to the
recoger
people who
información
subscribe.
y
And
cada
these
uno
are the
se puede
number of
apuntar. Estos son el
people
número de
registered with
personas que
cystinosis over the
hay
years. There will be
registradas
some
con
patients on this
cistinosis.
that have
Algunas
died
de
because this
estas
isn't
personas
censored
ya
for
habrán
death,
muerto porque
but
no
it gets to about
figuran,
180
pero son
odd
como unas
at the latest
180
count on
según
this one.
el último recuento.
And
No se
then
dan de baja
when
las
people
personas
come to
una
this
vez
hub clinic,
fallecen.
we've got some
Y luego
pages where we
también
enter data
hay
into RADAR.
páginas para poder entrar en el
So we talk
radar en
about
este
urine
sistema.
output and the
Y
gastrointestinal
hay aquí
system,
información
musculoskeletal,
sobre cuáles son las
and everything else. So this data then
afectaciones,
all
si
goes
son
into RADAR. And
gastrointestinales,
then in the long term, we'll have
musculoesqueléticas,
some really
etcétera, y
great
eso nos
follow-up
permitirá
data.
tener buenos datos de
It's broadly
seguimiento
mapped to
más a
all
largo plazo.
surfaces,
También se
European
vincula con las
database.
bases de
So
datos
there should be an
europeas,
ability in
etcétera,
the future
etcétera, y en el futuro
to put it all
tendríamos que
together.
poder agruparlo todo.
And so, as I mentioned, we
Creemos
think
que
hubs
estos
should
centros
be
de referencia deberían ser
accessible to
accesibles
all
para
patients.
todos los pacientes y
And we work
hemos
together. We can
trabajado
always work
juntos,
together
siempre
better. We're always
podríamos trabajar
very
juntos
busy, but we can always
mejor,
work
todos
together
estamos
a bit better.
siempre muy ocupados, pero vale la pena colaborar.
That's not a problem.
Hemos compartido
learning,
aprendizajes
quality
clínicos,
control and
sistemas de
improvement
control
and
de
research. We've heard
calidad,
a lot about today, which
investigación,
is
todo
brilliant.
esto que estamos
And
haciendo
we can
ahora también.
definitely try and move
Y
some of these
nosotros
things
también
forward all
hacemos
together
lo
in the
mismo entre
three
los
main
tres
hubs. And
centros de
also,
referencia
those of
y
you that are
algunos que no
not in
estén
the
en el
UK
Reino
will
Unido.
probably, hopefully, be
Espero
under
que
a specialist
tengan
hub in
un
your
centro de
country,
referencia en su país, y
if
si
you've
lo
got one,
tienen
and
y
we can work
podemos
together
colaborar
with the
con
hubs
ellos,
in
también podemos
Germany,
hacerlo, desde luego, porque
France,
tenemos contacto con
Holland,
algunos de los centros de
Belgium,
referencia de
etc.,
Francia,
to
Alemania,
try and
Holanda
improve
para
the care
mejorar
for
la
everybody.
atención que reciben
This is
todos, y
the
este
link
es
to the
el
website, as I say, just put it
enlace
into
a la
Google
web,
and find
lo
it.
pueden poner en Google, nos encontrarán, y con
Yeah, so
eso
that's
ya
my
termino.
last slide. Thank
Muchísimas
you
gracias
to
a todos.
everybody.
Y
Thank you. This
con
is my,
eso,
the adult team,
este es el
pediatric
equipo
team
de
at the Evelina,
adultos y el equipo
my colleagues in the
pediátrico
other hubs,
y compañeros de otros centros,
international
compañeros
colleagues,
internacionales de
pharmaceuticals,
las empresas
Cystinosis
farmacéuticas,
Foundation UK, CNE, and the
a
conference
CNE, a la Fundación de
listening.
Cistinosis del Reino Unido y a los organizadores de la conferencia. A todos debo darles las gracias por haberme invitado.
Thanks very much,
Muchísimas
David.
gracias,
The hard
David,
work
por este
has
arduo trabajo.
paid off. Can I just ask
Creo
a
que
quick,
como mínimo
do
ha
a
dado un
quick
buen
survey
resultado.
of
Quisiera
the
preguntar en la
audience,
sala
of those
de
patients
los
who
pacientes
live
que viven en Inglaterra,
England, are there any that have not
¿alguno
been seen in a
no
hub?
está asignado a un centro de referencia?
Okay, so that's really
Eso es muy
helpful.
útil.
That's a very quick win. We can solve
Eso
that
lo vamos a poder
quite
resolver
quickly. So come and talk to
fácil.
David
Hablen o
or
con
I,
David
and we'll
o
get
conmigo
that
y eso lo
sorted out.
resolveremos.
Any questions for
¿Alguna
David?
pregunta que quieran dirigirle a David? ¿Sí?
Yeah. The microphone is coming.
Ahora le llega el micrófono. Sin micrófono no podemos escucharles ni traducirles.
Hello. First of all, I just wanted to say, I think my experience with the hub, I attend your cystinosis hub.
Centro
I think
de
it's a great
referencia
concept, and as
es
it's
muy
been
buena,
developed,
es un
I think it's going to
concepto
continue to improve the
magnífico
life
que
of
ayuda a
people with
mejorar la vida
cystinosis
de
and having a
los
kind of
pacientes
one-stop
de
shop for
cistinosis.
things.
Esto de tener ventanilla única es
When you
fabuloso. Gracias.
mentioned about dentistry
Ha
and
hablado de
dermatology,
odontología.
I did wonder
Yo me
because
pregunto,
I was contacting someone in
yo hablé con
America
alguien
a few
en
years
Estados
ago,
Unidos
and I
hace unos
know
años y sé que
some
algunas
people with
personas con
cystinosis get
cistinosis
kind of, it seems like
parece
skin, like almost like an acne
tienen como
scarring
casi
type look, and I spoke to him. He said that was
como cicatrices de acné, y dijo que tenía que ver con la
obviously, I know a lot of
cisteamina en la piel.
transplant people see
Sé que hay
dermatologists in
muchos
regards to
pacientes
sun
que ven
protection,
al
but I
dermatólogo
wondered
para la
that, and also with
protección contra el
dentistry,
sol. Y
I've heard that there can be issues with the dentistry due to
también
some
he
of the
tenido
drugs that we
problemas
take
dentales
or
por
some of the
los
side
efectos
effects of the
secundarios de la
condition.
enfermedad
Is that
o por
something that
algunos
could
de
be
los
looked at in
fármacos que
future?
tomo. Esto no se
Including
podría contemplar de cara al futuro para incluirlo también en el centro de
Thanks, Avril.
referencia.
I'll start with dentistry because it's
Lo
so
de la
difficult. I
odontología
mean,
es muy difícil.
anybody trying to see a dentist dentist in the UK
Si
knows
uno
how
intenta
very
ver a
difficult
un
that
dentista
is.
en el Reino
I
Unido,
think
ya
we just
sabe
felt
que
that was a little
esto
bit of a bridge
es
too far. And also, we don't
complicadísimo.
really have any sort of
Nos ha
control over
parecido que
that.
no
You see what
podíamos
I mean? You know,
meternos en esa camisa de
we can
11
kind
varas.
of
No tenemos mucho control en eso.
persuade,
Podríamos
cajole, and bribe our
intentar
colleagues to, or pay our colleagues to come
pedir
to
a
the
los
clinic,
compañeros que
but
vengan a la
we don't
clínica,
really
pero
have
es
a
que no tenemos ni
facility
tan
to do
solo
that
las instalaciones para
with the
hacerlo
dentist,
con
unfortunately.
los dentistas,
No, I
con lo cual a
definitely hear
ellos
you, but
no les podemos ni
in terms of
invitar.
the hub, I think
Eso
that's
es
pretty
difícil
tricky.
en un
And
centro de
even
referencia de
within
los nuestros,
my
ni en
hospital,
mi
Guy's
hospital,
Hospital,
el
which
Guy's, que es
is
uno
one of
de
the
los
largest dental
que
schools in
tiene
the
una
UK,
facultad de odontología más
just even
grande
getting into that
del
can
Reino
be really
Unido, es
difficult for
difícil
anybody.
incluso llegar
So,
hasta ellos,
I hear you,
Así
but
que
it's
le
a really
comprendo,
big
pero es un
problem.
problema muy grave.
Dermatology
Y
is
la
an interesting one,
dermatología
isn't it? Because
es
there are
compleja.
some
Hay
cystinosis-specific
algunos problemas
issues,
específicos relacionados con la cistinosis, pero
but yeah, I don't quite know how to make that
no
work,
sabría muy
because
bien cómo
I think
conseguirlo.
because what's the
Porque la
intervention, you see? So
intervención,
if you've got this particular
si uno tiene un problema
issue,
concreto,
what
¿qué
are
va a
you going to
hacer
do
con
about it,
ello?
and
¿Quién
who has
tiene
that
los conocimientos de
expertise?
expertos?
I'd be interested to hear from
No
anybody
sé,
else
si
who
alguien
knows the
sabe la
answer to that.
respuesta a esta pregunta, encantado de que me echen un cable. Micrófono, por favor.
Yeah, as usual, you make a very good
Sí.
point, Abu. Yeah, thank you.
No hemos escuchado el comentario.
David, just to add to your point, and thank you, Avril.
Gracias.
So I help to run the clinic at Birmingham
Yo estoy en
Children's
la clínica
Hospital.
del Hospital Pediátrico de
And for
Birmingham.
dental and dermatology,
Para dermatología
although
y
we
odontología,
can't, get those specialties
no
to come to
conseguimos
a
que vengan los
clinic,
especialistas a la
we
clínica,
do
no,
try
pero
and,
intentamos
advocate for our patients that
que
have those
los
those
pacientes que
issues.
tengan estos problemas
And
podamos
we
derivarlos.
try where we
Siempre
can
que
to get
podemos
them into
intentamos
a
que
clinic
entren
pathway.
en las clínicas. De momento no tenemos mucha lista de espera, tenemos suerte. Y también hay algunos ámbitos en los que podemos llegar a estos servicios, pero es complicado. Y si empezamos a ver patrones nacionales, como pasa con otros temas a escala nacional, como BPS, y cuando los pacientes empiezan a darnos feedback sobre esto y a opinar, eso nos ayuda también a poder pedir a otras especialidades que vengan a ayudarnos. Así que pídanlo, no dejen de pedirlo, está muy bien que lo hagan.
Thank you very
Gracias
much
por
for
esta
the presentation
presentación.
and great work. This is really, really
Ha sido-
incredible. I wish we had this where
Ojalá
I was
tuviéramos
at
esto en
in the
Estados
States.
Unidos.
I was curious just thinking about, like, if you were to try to do something like this in the States, how
¿Hasta qué punto
difficult
fue
was it to
difícil
recruit
conseguir
non-nephrologist
que
specialists
los
to
no
come to the
nefrólogos
clinic?
vinieran a la clínica?
Many of them already have
¿Tienen
expertise
ya
with
experiencia en
cystinosis, or are many of them
cistinosis
learning as they
o van
join
aprendiendo a
these
medida
hubs?
que se añaden a los
Variable.
centros
So,
de referencia?
speaking
Bueno,
for
pues
Graham,
esto
his unit
varía.
already had quite a few people
Graham también sé que
in your clinic, didn't you, Graham?
le ha
Whereas
llegado mucha gente
for
en
me,
su clínica. La
most of
mayoría
my
de los
recruits, some of whom you would have heard yesterday, were fairly new to
que
cystinosis,
tengo
but
en mis
however,
centros son
sort of
bastante
academics
nuevos,
with
o son
interest in
gente que
similar
no tiene mucha experiencia en
conditions.
cistinosis, pero sí que hay muchos
So that's a
académicos que
fair point.
tienen experiencia en otras enfermedades similares. Así
I
que
think
la
it
observación
raises
es
a really important
importante.
issue,
Sí,
which is that,
es un tema
as David
importante,
said,
tal
we
como ha
had
dicho
some
David.
links, but it was fairly informal.
Teníamos
and
vínculos,
to
pero
make
era
this really
informal.
work, it has to be properly supported, funded, organized.
Y se
Otherwise,
necesita
it
apoyo
lasts
de la
for a while, and
organización,
then
porque si
somebody
no,
retires and it all falls apart.
pues
So
se
I think
necesita un
what
apoyo de la
David
organización,
said
porque
is
si no, a
make
veces
sure
cuando
that
uno
this
se
is
jubila,
set on
pues
a
ya se
permanent
desmonta
footing.
todo. Hay que hacer que las estructuras sean un poco más
I've
estables.
got a couple of points. So one's a question and one's a dose of irony. So the first one's about scripts. You mentioned you can get
Sobre
orders for Cystagon and Cystrops done
los
from the hubs.
pedidos que se
I
pueden
didn't really
hacer de
realize until Tina
Cistagón
mentioned last night that in England, I kind of have
y
to
que
be paying for
te las
scripts
pueden enviar a
in England.
casa.
We don't do that in Scotland.
En Escocia no,
So
no lo
does
hacemos,
that
no sabía que
negate
fuera
any
posible.
cost element
¿Implica
by doing that through the hub just for
algún
even those
coste adicional si se hace a través del hub?
that help patients?
I'm not sure.
Pues no estoy seguro.
So Does anybody
¿Alguien
know
lo
about
sabe?
does the home
Si el
delivery
servicio de
service
entrega a
have to
domicilio se
pay a
tiene que
prescription
pagar
charge for the home
aparte.
delivery service? I'm not
Lo
sure I even
siento,
know the answer to that.
podría saberlo, pero no lo sé.
I would imagine most
Creo que la
patients
mayoría de
who
pacientes
have
que
got multiple
tienen
prescriptions would have
muchas
the
recetas
what's it called, the
tienen
prepayment
aquello de
card,
la
prepayment
tarjeta de
certificate, which would hopefully
prepago que
mitigate
les
some of that
ayuda a
cost, because you'd be on
reducir los
lots of
costes
different
de
medications.
tener que recibir tanto medicamento. No lo sé.
One to think about.
Okay, thanks. That's really great. We'll take
Sí,
that away. Thank you.
lo apunto.
And the second point was about the situation we've got in
Sobre la
Scotland.
situación de
So, as you
Escocia.
say, we're served brilliantly by Glasgow and out in Edinburgh. And
Glasgow,
stuff.
Aberdeen y Edimburgo.
As we're applying for Persispi through the Scottish Medicine Consortium,
Nosotros estamos haciendo una petición a través del consorcio de de la medicina de Escocia. Se nos puede considerar una enfermedad huérfana o incluso ultra huérfana, pero para eso se tienen que cumplir una serie de condiciones como el número de pacientes, etcétera. No sé cuáles son las cosas, pero hay una que se me quedó grabada, que es que tiene que haber un centro de referencia nacional, y yo pensaba, en Escocia no lo tenemos, porque es tan escaso, es una enfermedad tan rara que es que ni nos plantearíamos que hubiera un centro de referencia en Escocia, pero en cambio se nos podría considerar, no se nos puede considerar ultra huérfanos si no lo tenemos, así que yo me he quejado, he escrito, pero no sé cómo demostrar que no tiene lógica que pongan estas condiciones. Tienen un centro de trasplante de hígado y de corazón. Ahí sí que tienen, ¿eh? Pero podrían plantearse que se llame el Centro de Designación Nacional, no sé, o el Centro de Referencia Nacional. No sé cuál sería el requisito burocrático para cambiarle el nombre, pero a mí me parece que si tienen uno, pues tienen que llamarlo así, como centro de referencia, para que les entre. Bueno. Seguramente el Departamento de Salud del Reino Unido tiene una única estrategia sobre enfermedades raras. Valdría la pena hablar de la cuestión. Bueno, creo que nos hemos quedado ya sin tiempo, así que me disculpo. Podemos seguir charlando durante el descanso. Lo que hemos visto han sido unas esperanzas maravillosas que se abren de cara al futuro para la mejora de la atención y del tratamiento de la cistinosis en distintos ámbitos. David nos ha contado cómo se está organizando la atención y cómo se pueden mejorar los resultados. Y eso incluye también investigación en estos centros, porque por primera vez ahora hay una masa crítica de pacientes que llegan al mismo centro, a esos pocos centros, y eso permite facilitar la investigación. Así que la clínica y la investigación muchas veces van de la mano. Bueno, acabando, quiero dar las gracias a todos los oradores por estas magníficas contribuciones, a todo, por estas charlas estupendas y por mejorar las perspectivas de futuro. Gracias.